Honke 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 riro, honke jombe jombe riro, honke 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 riro, honke jombe jombe riro.
The first international seminar on smart molecule of natural resources 2019 in Malang, Indonesia.
Bismillahirrahmanirrahim. The SMART 2019 officially open.
With the more sophisticated instrument in Thailand, so
when you tell science, your physical content is so low because for some reason your silica will also evaporate in a different form.
Mereka melihat bahwa AFOB is actually inducing apoptosis and killing the cells via apoptosis.
Associate the interaction between these two domains that make this domain start to convert and change the conformation.
Farnesene with C15, so the molecular structure is in the dark side, so sometimes it's difficult to understand it.
there's some studies that has the conflicting results about the treatment will be given as that, so not the dose of SSM.
ISSMART is the first international seminar on smart molecule of natural resources. Mohammed Answer!
Temanikan di berjoget sembang bertemu menangis membengkak jula-jula. Sila temanikan di berjoget sembang bertemu menangis.
And for C double bond,
profiling with the more sophisticated instrument in Thailand. So
when you tell science, your physical content is so low. Because for some reason, your silica will also evaporate in a different form.
Mereka melihat bahwa AFOB is actually inducing apoptosis and killing the cells via apoptosis.
Associate the interaction between these two domains that make this domain start to convert and change the conformation.
polymethyl methacrylates and due to degradation.
Farnesene with C15, so the molecular structure is in the right side, so sometimes it's difficult to understand this.
there's some studies that has the conflicting results about the treatment will be given as that, so not the dose of SSF.
ISSMART is the first international seminar on smart molecule of natural resources. More knowledge, answer!
Si batu manikan di pajoket sama di pertam dia menyanyi sebab mengkajula-jula, sebab mengkajula-jula. Si batu manikan di pajoket sama di pertam dia menyanyi sebab mengkajula-jula, sebab mengkajula-jula. Cuplak-cuplak suang eteng kelenter membuka tondong kudel pak empong lerak-lere sopong guyung dele ake sir pong dele kopong sir pong dele kopong. Ampar pisangku belum masak sabigi diurung bari-bari manggalepok patah kayu pengkok dimakan api apinya cangcurupan. Rasa sayangi lihat dari jauh. Rasa sayangi lihat dari jauh. Rasa sayangi.
Honke riro, honke jombe riro, honke riro, honke jombe riro.
Manikan di pajoket sama di pertam dia menyanyi sebab mengkajula-jula, sebab mengkajula-jula. Si batu manikan di pajoket sama di pertam dia menyanyi sebab mengkajula-jula, sebab mengkajula-jula. Cuplak-cuplak suang eteng kelenter membuka tondong kudel pak empong lerak-lere sopong guyung dele ake sir pong dele kopong sir pong dele kopong. Ampar pisangku belum masak sabigi diurung bari-bari manggalepok patah kayu pengkok dimakan api apinya cangcurupan.
the first international seminar on smart molecule of natural resources 2019 in Malang, Indonesia.
Bismillahirrahmanirrahim. The SMART 2019 officially open.
Oke, Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh.
Warahmatullahi wabarakatuh.
Her Excellency, the Vice Rector One of Brawijaya University, Professor Aulani Am, Honorable Professor Takeshi Omasa,
Recording in progress.
from Asian Federation of Biotechnology, Honorable the ISSMART Chairman, Professor Fatia, Honorable the ISSMART keynote speakers, respectable the ISSMART invited speakers, all ISSMART speakers, and attendants. First of all, let us praise and thanks to Allah Subhanahu wa Ta'ala, who has given grace and guidance to all of us. Therefore, we can gather in this occasion in healthy conditions. Salawat and salam may always be given to our Prophet Muhammad Sallallahu Alaihi Wasallam, who has guided us from the darkness to the lightness. Good morning, ladies and gentlemen. My name is Yorafika Dwi Wibangga, and I will be your Master of Ceremony for today's ISSMART. Welcome to the third International Seminar on Smart Molecule of Natural Resources, ISSMART, Asian Federation of Biotechnology 2021. This year's theme is Smart Molecules for Human Health and Prosperity. Previously, I would like to remind you, when you join this mind room, please rename your name into room A or B or C _ full name, since you will be assigned to join breakout room after plenary speech. Also, we will distribute the attendance list in the meeting chat around 9 a.m. Please fill the form out. Thank you. Ladies and gentlemen, ISSMART 2021 will run for 2 days, the 25 to 26 August 2021. The conference schedule for today is opening ceremony, keynote speaker speech, awarding certificate for keynote speakers, break, invited speaker speech, and will be closed by oral parallel presentations. Before we begin, let us spend a few seconds in order to pray together. Therefore, ISSMART 2021 will run smoothly. Bismillahirrahmanirrahim. Amin. Thank you. I would like to kindly request all ISSMART attendants to stand and sing to Indonesia National Anthem, Indonesia Raya. Thank you. For the first opening speech, I would like to invite Professor Takeshi Omasa from Asian Federation of Biotechnology. For Professor Takeshi Omasa, time and screen are yours.
Okay. Good morning, everyone. My name is Takeshi Omasa, professor of the Osaka University, and also I am the Deputy Secretary General of the AFOB and Chair of Biopharmaceutical and Medical Biotechnology Division AFOB. So thank you very much, and also the congratulations for their starting for third ISSMART meetings. Asian Federation of Biotechnology is established Asian network between some researchers. I think that everybody enjoys this meeting, and also there are well discussions. Well discussion make some very well networks between the researchers. Thank you very much, and also the enjoy meeting. Thank you.
Thank you, Professor Takeshi Omasa. For the next welcome speech, I would like to invite ISSMART 2021 Chairman to deliver a welcome. For Professor Pasia, time and screen are yours.
Good morning. Assalamualaikum warahmatullahi wabarakatuh. The Honorable of Rector Brawijaya University, Professor Dr. Insinyur Nufil Hanani, the Honorable of Vice Rector for Academic Affairs, Professor Dr. Hewan Aulani Amdes, the Honorable of Head Research Institute of and Community Service Universitas Brawijaya, Dr. Insinyur Bambang Susilo MS, the Honorable of President AFOB, Professor Wenchin Lee, PhD, the Honorable all keynote speaker, audience, and committee. On behalf of the organizing committee, I would like to welcome all of you, keynote speaker, distinguished guests, and participants to the third international seminar on smart molecule of natural resources, ISSMART 2021. For this year, the ISSMART 2021 held as a joint conference organized by Research Center Simona James and AFOB, or Asian Federation Biotechnology. Therefore, I believe that ISSMART 2021 will be an extraordinary event. The ISSMART 2021 ran for 2 days, August 25 and 26, 2021, as a virtual conference due to the outbreak of coronavirus. However, I believe that excitement to participate in ISSMART 2021 will not be reduced. The theme of ISSMART this year is Smart Molecule from Natural Genetic Resources for Human Health and Prosperity, which cover a broad range of research fields with various scope, such as biochemistry and molecular biology, molecular science, biomedical engineering, neuroscience, nutrigenomic, and nutrigenetic. The particular theme is intended to promote recent advance in the field of natural genetic resources, important to address important issue in human health prosperity. The International Forum also provides platform where nation and international academia for researchers, policymakers, and other stakeholders to translate technology, exchange ideas, and help stimulate multidisciplinary international collaboration in convergent manner for shaping a worldwide sustainable development. The selected paper from conference will be published in Journal Physics EOP Conference Series Journal Science and Technology Indonesia, and also Cismatex. Last year, we have published selected paper from second ISSMART 2020 in Journal Physics IOB Conference Series, volume 1665. Ladies and gentlemen, around 20 people registered to the seminar. The consists of 55 presenting participants, 6 keynote speakers, and the rest are non-presenting participants. The presenting participants come from Indonesia, university like Brawijaya University itself, Sahkuala University in Aceh, Sumatra, Airlangga University, Institute Center Research Indonesia, LIPI, and also Khairun University in Maluku Islands. In this meeting, we have presenter from overseas also. The student and researcher from Kyoto University, Niigata University, and a lot of student and researcher from Tsukuba University, and also University Putra Malaysia from Malaysia, and Suranare University of Technology from Thailand. Ladies and gentlemen, in this meeting, I would like also thanks to keynote speaker, Professor Renu Watwa from School of Integrative Global Major Sigma, University of Tsukuba, IS India Diverse Asset and Application International Laboratory, IS Japan, and also for Dr. Hendra Kuno Sewono, School of Pharmacy and Biomedical Science, Curtin University, Australia, and Associate Professor Mariana Kotudatken, PhD, from School of Biotechnology, Institute of Agriculture, Suranare University of Technology, Thailand, Professor Chi Taiyeh, PhD, from Taipei Medical University, Huangho Hospital, Taiwan. This is as a AFOB member, and Dr. Priyatna Sasmita is from Center Research RISE from Ministry of Agriculture, Republic Indonesia. And the last one is myself. The ISSMART 2021 could not become a reality without the help and assistance of many parties. Thus, in this occasion, I would like to sincerely thank to Rector Universitas Brawijaya, Head of LPPM UB Research Center Simona Kenes, and all members of the organizing committee. We have provided meaningful help assistance for the implementation of this conference. At last but not least, I would like to thank all of the conference participants who will contribute to making this most memorable event of third ISSMART 2021. We have tried to do our best to prepare ISSMART 2021. Nevertheless, there is nothing completely perfect in the world, including this conference. Therefore, please accept our deeply apologies for any inconvenience found in this conference. Please enjoy ISSMART 2021. Wassalamualaikum. Good morning. Enjoy your seminar.
Thank you for Professor Fatia. For the opening speech, I would like to invite Vice Rector 1 of Brawijaya University to deliver an opening speech. For Professor Aulani Aim, time and screen are yours.
Good morning, everyone. Assalamualaikum warahmatullahi wabarakatuh. On behalf of Universitas Brawijaya and organizing committee, I would like to extend my warmest welcome to all delegates of the third International Seminar on Smart Molecule of Natural Resources, ISSMART 2021, in Universitas Brawijaya, where the theme of this conference is Smart Molecule for Human Health and Prosperity. Especially, I would like to express my deepest appreciation to all keynote speakers, Prof. Renu Wardana, PhD, from School of Integrative and Global Major, University of Tsukuba, Japan, Dr. Hendra Guno Sewoyo, School of Pharmacy and Medical Sciences, Curtin University, Perth, Australia, Associate Professor Marian Ketudat, PhD, School of Biotechnology, Institute of Agriculture Technology, Surani, University of Technology, Thailand. Prof. Chi Da-Ye, PhD, from Taipei Medical University, Suangho Hospital, Taiwan. Dr. Priyatna Sasmito, Pusat Penelitian dan Pengembangan Tanaman Pangan, Puslit Bangtan, Ministry of Agriculture Republik Indonesia. Prof. Fathia, MKes PhD, my friend, Head of Smenogen Research Center, Universitas Brawijaya. I would like also to take this opportunity to express my gratitude to the conference committee for organizing the seminar today, ISSMART 2021. Ladies and gentlemen, due to this pandemic, Universitas Brawijaya adapts in every aspect, both in academic and non-academic activities. As part of our adaptation in academic activities, we have arranged online lecture and discussion, also virtual international conference since the beginning of the COVID-19 era pandemic. This international seminar, ISSMART 2021, also provides a platform for national and international academia, for researchers, policymakers, and stakeholders to translate technology, to exchange ideas, and to have stimulate international collaboration. This international seminar is also one of the forms of our adaptation in this pandemic COVID-19. Ladies and gentlemen, as we know, the COVID pandemic is still ongoing. The COVID-19 pandemic has deconstructed various dimensions of life, the changing of our lifestyle. So this seminar, ISSMART 2021, is going to be held as a virtual seminar. I know you all from various places who still meet virtually in this seminar to enrich our insights and knowledge. In addition, collaboration between Universitas Brawijaya in other universities, both from Indonesia and overseas, can be continued. I'm sure this conference should bring advantages for all participants. They will learn many new aspects of research in related topic, either from keynotes and invited speaker or general participant as well. Ladies and gentlemen, in this occasion, I would like to sincere thanks to Head of Smenogen Research Center, Prof. Fathia, and all members of the organizing committee who have provided meaningful help and assistance for the implementation of this seminar. Today is an especially great moment for us to have such world-class speakers and distinguished participants for our conference. And last but not least, I would like to thank all to the conference participants who will contribute to making this the most memorable event. Again, welcome to this conference. And please have a nice enjoy this international seminar ISSMART 2021 with a great beneficial for us. Thank you very much. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you for Professor Aulani Am. For the next schedule is plenary speech. Our keynote speakers for this morning are Professor Renu Wadwa, PhD from School of Integrative and Global Majors, University of Tsukuba, AIST India Diverse Assets and Applications International Laboratory, National Institute of Advanced Industrial Science and Technology, AIST Japan. Associate Professor Mariana Ketudat Keynes, PhD, from School of Biotechnology, Institute of Agricultural Technology, Suranare University of Technology, Thailand, and Dr. Hendra Guno Sewoyo from School of Pharmacy and Biomedical Sciences, Curtin University, Perth, Australia. The moderator of plenary speech is Anna Safitri, PhD. Before, I would like to deliver the curriculum vitae from Anna Safitri, PhD. Yeah, Anna Safitri, PhD. She works at Chemistry Department, Brawijaya University, and Research Center, Smona Genes, Brawijaya University. She have studied at Brawijaya University, the University of Sydney, Australia, and then to take PhD at the University of Sydney, Australia. She is professional membership of Himpunan Kimia Indonesia, Perhimpunan Biokimia dan Biologi Molekuler Indonesia, Royal Society of Chemistry, ASEAN Federation of Biotechnology, and Korean Endocrine Society. And please welcome Anna Safitri, PhD. Please, time and screen are yours.
Thank you, Yora, for the introduction. Good morning, everyone. My name is Anna Safitri. I'm from Brawijaya University and also from Research Center Smart Molecules of Natural Genetic Resources, Smona Gens, that is the organizer committee for today's event. For this morning plenary session, we will have three great keynote speakers. They are Professor Renu Wadwa from Japan, Associate Professor Mariana Ketudat Keynes from Suranare University of Technology, Thailand, and Dr. Hendra Gunoswoyo from School of Pharmacy and Biomedical Sciences from Curtin University, Australia. Without further ado, I would like to introduce the first keynote speaker for this morning. She is Professor Renu Wadwa, PhD from School of Integrative and Global Major, Sigma, University of Tsukuba, AIST, Japan. The operator, please can you share screen? Professor Renu Wadwa CV? Yes. Okay, ladies and gentlemen, this is a short CV from Professor Renu Wadwa because I believe she has more than this achievement. So for Professor Renu, she graduated her PhD from Guru Nanak Dev University, India, and PhD from the University of Tsukuba, Japan. And then she continued her postdoctoral in the University of Newcastle, England, and Duquesne, Japan. And she also has more than 200 publications in international peer-reviewed journals with many invited or plenary talks in international conferences with the high Scopus index of 50 and high Google Scholar Edge index 63. And she has quite so many academic propositions here. So you can see that she is become professor in School of Integrative and Global Major, Sigma University of Tsukuba, Japan, and also many academic position that is really prestigious. So we really have very lucky to have Professor Renu Wadwa this morning. And also I would like to invite Professor Renu to deliver her talk. And because we already have her video presentation, we will play her video presentation, but Professor Renu Wadwa is here. She is here online with us, and she's ready to answer and to take all your questions in discussion session.
Would you like me to speak or would you
This
like
is,
to play?
Professor Renu Wadwa presentation.
I can play from here.
Hello, everyone. Very good morning. And first of all, thank you organizers for the invitations. It is my pleasure to be with you on this third international seminar on smart molecule and natural resources. My name is Renu Wadwa and I'm reaching you from Japan.
So, Professor Renu, are you here?
Yes.
Public institution.
Yeah.
And I'm a prime senior scientist at
speak from
and
here.
also professor. Today, I'm sharing some of our research results with you on the topic, as you see on my slide, experimental evidence to the nutraceutical and pharmaceutical potential of an Indian Ayurvedic herb, Ashwagandha. Well, my lab for last 30 years has been engaged in understanding the molecular mechanisms of aging, stress, cancer, and neurodegeneration. Well, stress and aging, I'm sure we don't need introduction to these very universal phenomena, rather a fear. We all undergo aging and we all have stress. Rather, mild stress may indeed be good for you because it boosts up your defense mechanisms. So indeed, irrespective of where you are, what you do, what age you are, we all have stress. And stress as such may not be a big worry. But the stress-related pathology becomes a big worry, and that's where we need to deep. One of the pathologies I'm going to talking about in my talk today is cancer. As a simplest definition of cancer, it is uncontrolled proliferation of cells, and it can happen anywhere in the body. It will travel from primary site to the secondary sites in most of the cases, disrupts more normal body functions, multifactorial. And the mechanisms, if you want to broadly define those mechanisms, it could either be activation of genes that are involved in proliferation of cells, called oncogenes, or inactivation of the ones which inhibit proliferation or uncontrolled proliferation, that's tumor suppressors. And treatment, I'm sure you all are very well aware of, and also you probably also know the incidence of cancer is increasing rapidly globally. From somewhere in 40 million in 2012, it's expected to reach to 22 million in 2030. So it is rather a serious issue. Some common facts about carcinogenesis, that's the process or phenomena or the process of cancer. It could be, as I said, activation of proteins or inhibition of proteins, which are indeed tumor suppressor proteins. These cells attain autonomous growth without even the growth factors or the growth signals. And they become resistant to extracellular growth inhibitory signals, resistance to stress-induced cell death, and they start maintaining their telomeres, and stimulation of cell migration and neoangiogenesis is another phenomenon. Now, in the laboratory, in order to understand the process of carcinogenesis, we have been using the cellular model, using normal cells and cancer cells in parallel. Now, normal cells in culture dish can divide only for limited number of times, and they then stop dividing and enter a state of permanent growth rest that's called replicative senescence. So normal cells, just like us, age when they are cultured in the laboratory. In contrast, the cancer cells, they're immortal. That means they can go on forever and they can form a mass of cells in even the culture dish. So this unlimited number of divisions that the cells can go through is called immortalization, and that's what is a cancer type in culture dish. Now, very similar to carcinogenesis or cancer phenotype in culture dish, we can also recognize the stressed cells in culture dish. For example, by morphology, they can be seen, and also the whole lot of signalings get altered if the cells are stressed in culture dish. So, in other words, this provides a very unique and very powerful platform to study the phenomena of aging, cancer, and stress. For those of you who have not been doing the cell culture, this is how the cell culture laboratory or the incubators look like. Researchers culture cells in dishes, and if they're, as I said, they are normal, they will stop dividing. And if the cells are cancerous or they are derived from some cancer or tumor tissues, they can go on forever and they can be established into permanent cells, they're called cell lines. Now, using this platform over the years and years, I have been able to clone a new protein called morglin. I named it morglin. It is a member of HSP70 family of proteins, and there are over 150 publications on this protein describing its role in carcinogenesis, human carcinogenesis, how it inactivates the tumor suppressor protein P53. The second protein that was cloned in my lab was also related to regulation of P53. We named it CARF for collaborator of ARF. And many of you may know that ARF is a protein which regulates P53, which is a guardian of the genome. It's a major tumor suppressor protein that regulates controlled proliferation of cells in either the normal environment or abnormal or stressed environment. Bringing all these research results to society is a longer path, even difficult than doing results and producing publications. Now, there could be two ways that we can bring these research results to society, either by pharmaceuticals, have inhibitors or the drugs which will regulate these proteins, or the nutraceuticals. As you would all acknowledge that pharmaceuticals is a longer and difficult path, requires lots and lots of money. And nutraceuticals, the knowledge that we build up on nutraceuticals can be available and used in society much more easily. So in my lab, we've been following both the paths. We've been building knowledge on the role of morglin and CARF in human carcinogenesis. Based on that knowledge, we've been also doing screenings for the compounds, the smart molecules, natural compounds, which are either inhibitor of morglin or CARF, and they can regulate the process of carcinogenesis. Here I list some of the lead compounds from my lab, and there's a lot of research has been done and published on ashwagandha leaves, and today I'm going to describe on these. There are other smart molecules or the small molecules also recognized and identified as morglin inhibitors and candidate potential compounds for treatment of cancer. Right, for ashwagandha, for those who do not know this herb, this is a proud herb of Indian Ayurvedic traditional home medicine system. Its botanical name is Vidania somnifera, and it's been used for supplement, for enhancing quality of life, for sleep, for a variety of ailments in India and neighboring countries, rather Asian continent, for over 3,000 years. So when we wanted to start with some herbs, we picked this up as a queen of Ayurveda from the literature, and we began to look at the mechanisms of action. And our first, tasks were to see if it has anti-cancer, anti-stress, or anti-neurodegeneration activities by using our favorite models on, research. Here I'm showing you what we did. We prepared extracts from ashwagandha either by alcohol or water-based, identify the compounds that are contained in these extracts, and then we put these extracts on the cellular models. For example, we are using cancer model, we are using premature aging model, industrial metabolite stress, and neurogenesis, and then take these results from cell culture system to also animal models wherever it's possible. Now, for those who are not doing research directly on cancer, anti-cancer activity in animal models is usually studied by injecting mass of tumor cells underneath the skin of mice, and you let these cells grow into tumors, and you can study the effect of your compounds by either injecting them into the tail vein, or you can have the subcutaneous or local injections likewise. In our system, this was indeed done by my PhD student, Widudusan from Indonesia, Brawijaya University itself. And this has been a landmark result in this field of research results. What we found was that the local injections of ashwagandha alcoholic extract, we call it I extract, either the local injections or the oral feeding to the mice which bear tumors, was able to reduce the growth of the tumors very, very significantly, showing that it has a remarkable anti-cancer activity. We brought that to a cell culture system, and when we treated the cancer and normal cells in the culture dish, you can appreciate here that the cancer cells are dying or undergoing apoptosis. The normal cells are growing normally as they were in the control culture. So just a summary that the ashwagandha leaf extract, alcoholic leaf extract, was able to kill cancer cells selectively. So that was our first finding on this one. And then we wanted to know the mechanism of action of the selective anti-cancer activity. And we did a variety of modern technology assays. Here we are doing loss of function screening, where we knock down gene by gene and see if the effect, which is cytotoxicity here, is taken away by knocking down of certain genes, which would mean that these genes are critically important for bringing cytotoxicity of cancer cells. We did that for siRNA library. We also performed the unbiased randomized ribozyme library screening in a similar way. And then the targets that we achieved from these two parallel screenings, we put them through the computational analysis and see which signaling pathways were critically picked up there. And by this one, we found that the activation of tumor suppressor protein P53 was highlighted in these screenings as a predominant mechanism of apoptosis or growth arrest of cancer cells induced by I extract. Now, this research is all published, so I wouldn't be showing you all the data here, but just as a summary of this, what we have published is that the ashwagandha anti-cancer bioactive ingredients, bidafrin A and bidanone, are able to abrogate Mortlin P53 interactions. Initially, Mortlin P53 interactions keep P53 in the cell cytoplasm, and thereby P53 is inactivated. When ashwagandha bioactives are interfering with these interactions, P53 can go back to the nucleus and then perform its function in either the growth arrest or apoptotic signalings. So you can imagine that Mortlin is a girlfriend of P53. It keeps P53 in the cytoplasm, which is a home, and then ashwagandha acts as a mother or caretaker, separate them and let P53 go back to its job in the nucleus. And then the cells are put into growth arrest or apoptosis. Now, experimental evidence to this, as we have published in several papers, indeed, breast cancer cells, as you can see, the treated cells, the cancer cell, they're showing increased amount of P21, which is a downstream effector of P53 protein, very important in the growth arrest of cancer cells. So it's increased in cancer cells, but not in the normal cells. That amounted to selective toxicity of ashwagandha I extract to cancer cells. Similarly, we have looked into several signaling mechanisms. We have published that the ashwagandha I extract and its active components, bidafrin A and bidanone, they kill cancer cells by induction of oxidative stress, by inhibition of aurora TPX kinase, which is very important for cell signaling, By inhibition of HNRNPK, it kills the cell migration, and it also inhibits ALT, which is a crucial for telomere maintenance in telomerase minus cancer cells. In other words, meaning that such natural compounds can also be useful for treating very difficult cancers which do not respond to telomerase inhibitors. So this is all about the cell culture. I wouldn't be having much data on the mouse models or different kinds of cancers, but there's a lot on the online that can be seen. And one of the example is this patient who was benefited by ashwagandha therapy. I welcome you all to go through his history, which is available online. Now, we wanted to see that what I extract or its components do to the normal cells. So we started looking at, again, Vidudu had done this work. We started looking at normal cells and cancer cells in parallel. You are looking at three kinds of normal cells and cancer cells on the top. Treatment of these cells with eye extract, with the known, with Afrin A, and the combination is shown. And you can actually appreciate here that Afrin A kills cancer cells and was affecting the normal cells too. But here, with the known, the normal cells were as normal as in the uncontrolled, in the controlled cultures. So this meant the selective toxicity of eye extract to cancer cells and no effect to the normal cell. When we did the dose response assays, it turned out to be that with the known low doses, they are indeed good for normal cells. You are looking at cells which are stressed by oxidative stress, by hydrogen peroxide or UV, and with the known supplemented cells or their cells that are grown in with the known supplemented medium, they look much better and healthy here and here. The long-term serial passaging of the normal cells in the medium that has low amounts of vidanone led to their lifespan extension, and there were decreased number of aging cells or beta-gal positive cells in treated dishes as compared to control. All this data means that low or non-toxic doses of vidanone were able to cause lifespan extension and also protected the cells from oxidative stress. Now, again, the work was continued by my PhD student, Didik Priyandiko, who is back to Indonesia and leading his own research team in the university. So he did these stress assays where he stressed the cells with MAA. MAA is a toxic metabolite of esterapholates, and esterapholates are used all over in agriculture, medicine, plastics, sheets, in all plastics things. So we are exposed to MAA regularly. So when Didik treated the cells with MAA, you can see the toxicity, the decrease in cell number, increase in aged cells, increase in DNA damage, increase in oxidative stress, and decrease in mitochondrial membrane potential, which is mitochondrial damage. The cells treated with MAA plus vidanone were protected to a large extent. So vidanone was able to protect the cells against industrial, chemical, or chemical toxicity. Continued to this, we continued to look at effect of vidanone to the normal cells and in response to a variety of stresses. And here I'm showing you some data on the oxidative stress and old age pathologies. I'm sure you know that there's loss of memory, there's accumulation of oxidative damage, and there is accumulation of amyloid plaques in Alzheimer's brain. And there are chemical models to study this disease. There's an oxidative stress by hydrogen peroxide, glutamate stress, scopolamine, where we can actually induce memory loss, and MTTP, which can actually give you Parkinson's model of disease. My student Navjot Shah continued to look into these phenotypes and effect of eye extract and vidanone. And what she found was the cells, when stressed with all these kinds of stresses, they die with apoptosis. But when the known is there in the culture medium, the cells are protected and they also undergo differentiation into astrocytic or neurite morphology. I wouldn't show you much of the data, but all these beautiful extension that you see is the neurodifferentiation, which is caused by eye extract medium or the Vidonol medium. Another student, Arpita Konar, she took these assays to a mouse model. And here we are creating the loss of memory mouse by scopolamine. And what she found was that the loss of memory was protected by supplementation of eye extract. And also normally the unstressed mice also showed better memory when they were fed with the eye extract. In the Parkinson's model of disease, as you probably know that this Parkinson's model of mice, they have a wobbly walking as you see here. And if the mice are fed with eye extract, their walking is corrected to a large extent. So all this data means that ashwagandha is good for management of cancer, good for management of stress, and good for age-related neurodegeneration and brain dysfunction that I showed you for a variety of cell culture and animal models. Now, the most stress right now globally, we have new strain of coronavirus and COVID-19 pandemic. I'm sure you all know about this timeline starting from 2019 till today. Millions and millions of lives have gone into this, and all research resources are focused on fighting against this pandemic. So let's review how the virus infection takes place. Most of you might know that there is a attachment step, there is penetration of the virus to the cells, host cells, then the biosynthesis of the viral particles. It takes place by using the whole cell machinery. Maturation of these infectious particles or viroids takes place, and then it is lysed, and the infection goes from one cell to the next and next very, very rapidly. TIMPRESS2 and ACE2 here, ACE2 and TIMPRESS2, they play extremely important role in attachment of the virus to the cell membrane and its penetration to the inside to the cells. And once inside, MPRO and other proteins of virus, they are involved in synthesis or replication of the viral genome and making the infectious particles. Now, with this knowledge, and we looked at the effect of eye extract and a variety of other purified vidanolites as listed here on the expression of TMPRSS2, TIMPRESS2. And what we found that the treated cells, they have down regulation of TIMPRESS2 expression. So this means that the treated cells would offer some kind of blockage for to enter the cells. Similarly, we looked at ACE2 expression, and you can appreciate that the treated cells, they have low level of expression of ACE2 as compared to the control cells. So once again, if the both receptors are enter the cells. So these are the two cell surface receptor proteins. And Vidonol and Vidofludimus, along with the other vidanolites, we have tested, and we also see now with the computational analysis whether these compounds can dock into the active sites of these proteins, plus the MPRO protein, which is involved in viral inside the cells. We collaborated with, and we continuously do that collaboration with Dr. Durai Sundar from IIT Delhi. And here I'm showing you that we checked that the most of these vidanolites listed up here have capability to interact with ACE2. And looking at here, the ACE2 expression with these vidanolite treated cells, not only the protein, but also mRNA was decreased in cells. Similar to ACE2, we are looking at TIMPRESS2. TIMPRESS2 also showed the capability of docking to these vidanolides, and then looking at its expression level. With many of these vidanolides, we are getting that the expression level was decreased, not at the protein level, but at the mRNA level. Now, the obvious question is to see the real replication assays, and in collaboration with RCB, Regional Center for Biotechnology, India, we have done these assays, and what we have found here is that there is a decrease in viral replication in cells treated with vidanolides. Most effective was here vidanolide B, and then vidanoside 5, and then followed by vidanol. But when we use the extract, which provides you a complicated and a mixture of different vidanolides, we were able to get 38 to 90% inhibition of viral replication. Now, this obviously tells you that it's important to have extract or plants with a defined amount of active ingredients, which are vidanolides. Now, which ones of these would do which job is the research result. For example, vidafrin is most important for the cancer treatment. It has a top anti-cancer activity. As I showed you, vidanolide B and vidanol and vidanoside 5, they are better for COVID-19, and vidanol is good for stress and age-related pathology treatments. Now, taking that into mind, we've been collaborating with Guru Nanak University, India, and industries in Japan for making well-defined ashwagandha plants. And we have also established hydroponic ashwagandha, which takes much less time and it is more defined in its ingredients. I'm not sure if this video will work here, but anyway, so we've been able, this is all published, and we've been able to get hydroponic ashwagandha plants too. So information can be seen online. All these papers are published, and at the end, I'd like to say thank you to the people who did the work. Vidudu is here, Didik Priyandiko is here, and they initiated, when the ashwagandha work was just initiated in the lab, they were the ones who did the work. It's been carried out, and several Indonesia students have visited my lab, and it can still continue with the current students, and Anisa and Hasna. They are the current members, they're current students, and they're also doing some work on ashwagandha, on other smart molecules that are being studied in my lab. Thank you very much.
Thank you very much for Professor Enuwatwa for deliver her talk. For the next speaker, we will have Associate Professor Mariana Keynes, PhD from School of Biotechnology, Institute of Agricultural Technology, Suranare University of Thailand. The title of her talk is Sperm Sexing Technology Using Smart Molecules, Antibody Against Bovine Wild Sperm. For the committee, can you display Professor Mariana CV first? Okay, so here we have a short CV from Professor Mariana Ketudat Keynes. She's from, as I said before, School of Biotechnology, Institute of Agricultural Technology, Suranare University of Technology, SUT, Thailand. So for her educational backgrounds, she gained her PhD from University of California, San Diego, USA. And then here are some her job experiences. Right now, she's academic staff in Suranare University of Technology, Thailand. During 1988-1994, she was working in University of California, San Diego, USA. So she also has a lot of publication with 43 papers indexed by Scopus with its index of 16. Here are some of her awards. You can tell that she's got a lot of awards here. So we also very lucky to have Professor Mariana Ketudat Keynes this morning for our keynote speakers. Okay, so without further ado, I would like to invite Professor Mariana Ketudat Keynes to deliver her presentation. For Professor Mariana, would you like us to play your video presentation?
Yes, I think since I already had the video, you can go ahead. The video was not that great, but hopefully it will be okay. Please
Okay.
ask a lot of questions later on.
Thank you very much. So for the committee, I would like to share screen the video of Professor Mariana. Thank you.
Good morning, ladies and gentlemen. I would like to briefly introduce myself again. My name is Marina Ketudat-Kearns. I'm working at Suranari University of Technology, Nakhon Ratchasima, Thailand. And today I would like to tell you a little bit about my smart molecule. My smart molecule that I would like to talk to you about today is monoclonal antibody. And what do I use the monoclonal antibody for? I use it for cattle sperm sexing. Before I start, I would like to thank the organizing committee, especially Facia and her team, for inviting me to be the keynote speaker today in the 3rd International Seminar on Smart Molecule and of Natural Resources. So the topic for the meeting with Smart Molecule for Human Health and Prosperity, but I would like to move a little bit away from human health, but I would like to talk about cattle sperm sexing. So hopefully you will bear with me about the cattle sperm sexing. Okay. As you know, most mammals, most animals, the offspring will be 50% male, 50% female. If you want your baby to be female, oh no, what can you do? Can you do something? If you want the baby to really be male, is there something you can do? So for my work, I focus on cattle. Why cattle? If you are a dairy farmer, you would like to get milk from your cow. You would like to have female baby, correct? So that was what brought me into this work. So for cattle, baby or female heifer or young teenage cow, when they get in general at the age of 2, get to their being mature enough to have babies. So the farmer will synchronize, do the estrus, check the estrus cycle, and then mainly they will do some artificial insemination with some good donor sperm. Normally you have to do at least twice. When your baby cow get pregnant, you'll be happy if you get female babies so that you can do the milking and the process continue. However, it will take you 9 months for a baby cow to get pregnant and be born. If the baby is born male, what you can do? The farmer wasted 9 months waiting for a baby. You can sell the calf at a low price for barbecue. So, like I say, normally 50% male, female. Is there anything we can do to increase the number of female cattle? So the current technology, what does people do? Current technology can use flow cytometer to separate male and female sperm. However, these current technology separating male and female sperm, the sad part is the sperm are normally not really healthy and will be more difficult to get pregnant mother to give rise to female cat baby born. So our innovation is we have produced monoclonal antibody that at this stage we call it bull off. We produce monoclonal antibody with the procedure of sexing the semen or the sperm. For the frozen semen, we can use our product, and we can use the sperm that has been sexed in artificial insemination, or we can use the sex sperm in the IVF procedure, in vitro fertilization. After getting the sex embryo, we can do the embryo transfer into surrogate mother. So that is the overall of the work I want to present today. So our core technology, what have we done? We started off using sex sperm from the using flow cytometry to produce monoclonal antibody. This was done actually more than 10 years ago. So using sex sperm as an antigen, and then we got the spleen of the mouse fused with the myeloma cell, get the myeloma, screen for antibody that is specific to sperm, later on screen for antibody that's specific to Y sperm. And we got three clones of hybridoma that was able to produce antibody specific to Y sperm. So you can see in some of these pictures some sperm and the antibody staining the head of the sperm. After obtaining the antibody, what do? We develop our sexing product here called bull off, can mix with the sperm. This antibody is against the Y sperm. So Y sperm will be bound with the antibody. After that, we use some specific bomb to kill the Y sperm, leaving only the X sperm. And then we can use the egg sperm to fertilize with the egg, can do artificial insemination or do in vitro fertilization, and happily, hopefully, female calf born. The result in our laboratory for the normal sex sperm, 50-50 female, 50 female and 50 male. And for our sex sperm using our antibody, we got 80+% of female. And here is our unpublished data in the lab. We had 3 different antibody clones. We used these antibody to separate the X and Y sperm, and then we use the sperm to do in vitro fertilization, and we can see that the sperm treated with the antibody give the number of cleaved embryo. That means embryo that can grow pretty much okay, not too much different from the control. And afterwards, we checked the sex of the embryo. We found out that in the control, normally should be 50, but we got only 42% female. So maybe there's something about the sperm that we use in this experiment that there's more of the male sperm. But this sperm, after we treat it with our antibody, can increase the number of female between 78 to almost 87%. So we were pretty much happy with this. However, our funding ran out. So we stopped our work here. And one of our friends heard about this work of our antibody. Professor I.L. Kong, or we call him Professor King Kong from Korea, heard about our work. At that time, he was also working on Y-specific monoclonal antibody from Korea. And he published this work about using the Korean Y-specific monoclonal antibody to produce female cattle. So this work was published in 2019, just a little bit after he talked to us about our work. So at that time, he asked us to send some of our antibody for him to do the experiment comparing with the Korean antibody. So the work from Professor Kong's lab showed that our antibody was able to work well to aggregate the sperm. You can see if we use only 1 microgram or 5 microgram, not much effect on to the sperm. However, at 10, 15 or higher, we can see aggregation of the sperm, showing that, okay, this antibody works and the sperm can bind, come together. So the antibody, so he can show, so the work from Professor Kong's lab showed that 5 microgram of our antibody works well, or another, we had 3, but he was working on only 2 of our antibody, and the sperm viability still okay in both of the antibody used in this work. Comparing our antibody with the antibody from Korea that he was using, can see that our antibody can also work well, can aggregate the sperm, but we need to use 10 to 15 microgram of our antibody compared to the Korean antibody that don't really need to use that much. Using our antibody showing percent of blastocyst or development of the embryo, no different than control, showing that, okay, even the sperm treating with our antibody can aggregate the Y sperm, and the rest over of the sperm can be used for in vitro fertilization and worked well for the cell, the embryo to grow. The embryo that grew up was tested for the sex, and Professor Kong group found out that got 84% female from their first experiment. Their second experiment, 89% female. So if you are a farmer, you will be very, very happy. Not 50% male female anymore, but 89% female. You'll be pretty much happy. So for this work, since I'm in the field of biotechnology, we are not just doing basic science. Have to see, oh, can we really put our science up to work, up to the farmer, for the public? So I asked some of the students to come and help me do some business model and a lot of negotiation and doing a lot of designing business model and things. So our student got several of the prizes and award for Global Connection Life Science Cluster. So and also research to market award. And another group of people heard about our work. The company called Chromosexin in Australia found out about our work, and it purchases 1 milligram of our antibody for 7,000 US dollars. Professor Kong, who's working on sperm sexing in Korea, was very happy with the result, and right now he's purchasing already 5 milligram of our antibody with a secret price, but I was pretty happy about the price he gave us. So to compare our work, our result with some other technology in the market, our technology gives approximately 80% female using bovine anti-bovine Y sperm antibody. The quality of sperm is very high, and our price is much lower. So that is for, we still developing the method of how can farmer really take our antibody to use in the field. So we are still in the process of developing the process. But another idea came up, or actually farmer came to talk to us. We were working on increasing the female. Some beef cattle farmer came to us and said, hey, we want male because male cattle grows much faster. Can you produce male cattle for us from your antibody also? So that came up with another idea of work that we've been trying to do recently. Can we develop the process of selecting for Y sperm instead of killing Y sperm? So we try to attach the anti-bovine Y sperm antibody, the whole antibody that we have, to some magnetic beads and develop the process of sperm separation. So we were able to attach the sperm to the magnetic beads. So we hopefully, the magnetic can just pull the Y sperm and let the X sperm to swim away. In the past, we destroy the Y sperm to get the X sperm. In this case, we want to save the Y sperm. So we started off with producing a lot of the monoclonal antibody, lyophilized, dialyzed and lyophilized them. So we have a lot of antibody in stock right now that we already test, and it was well to be able to select for X sperm, but now we want to select for Y sperm. So here's some example of some frozen semen after wash. We have to wash to get rid of the mixture, mainly egg yolk that's in the frozen semen. So here's the sperm precipitate at the bottom. And after the reaction with our antibody, see there, and we repeated Professor Kong's experiment. Yes, we can see a lot of aggregation after putting our antibody. So we can see some aggregation of the sperm. Now we use the magnetic beads to attach to the antibody, or attach the antibody to the magnetic beads. You can see here the magnetic beads attached to the, and before attached to the antibody, And after attachment, and here's the mixture of the sperm and the magnetic beads. So we can see some clumping of the sperm. So far everything looks good. And we also develop the process of checking of the X and Y sperm by using real-time PCR. We develop the washing process of the sperm, the binding of the monoclonal antibody to magnetic bead, develop the real-time PCR to analyze the X and Y sperm. However, at the end of the day, we found out that our anti-bovine antibody attached to the magnetic beads can only separate the Y sperm only 59%. So this is the test from using real-time PCR. So we are not too happy with this result. However, we would like to develop and try maybe the washing process a little bit better and repeat this experiment again to hopefully able to really obtain the Y sperm. Before closing, I would like to repeat and tell you that we were able to separate X sperm up to 80 to 85 or even 89%. So if anybody want baby female cow, we can do it. Not a problem. However, this work is by destroying the Y sperm. But for cattle farmers that want beef, they want male cattle, male baby, we still in the process of developing. We still not able to do that work yet, but hopefully maybe in a year or two, we will have some good result to show you. Before the end, I would like to thank the National Research Council of Thailand, Northeastern Science Park of Thailand, and Office of Technology Licensing Technopolis at Suratnari University of Technology, TLO, for their support and funding. And would like to really thank Chromo Saxon company in Australia that bought our antibody at a quite high price. That was in the beginning. However, they were not able to give us any result yet from the antibody use because of this COVID crisis. And I really would like to thank the King Kong company or Professor Kong group that decide to really buy and use our antibody to produce their female cattle. Last but not least, all the work here would not be possible if not because of Dr. Surachai Ratanasuk. This was his PhD work more than 10 years ago to produce the monoclonal antibody. And Dr. Sumit Imsunthon Raksa, who recently got Surachai's antibody out to do the work and put it to public and put it out onto the market. So now Dr. Sumit has his own company selling these antibody. All this work will not happen if Professor Rangsan Pampai didn't ask me and Surachai to do this work long time ago. And also thank Professor Kong for helping with all the work and confirm that our work is worthwhile and should be out to the market. Thank you very much. Terima kasih. And now I'm open for question and comment. Thank you.
Thank you very much for Professor Mariana Katudatkin for the interesting talk. It's so interesting about talking about cattle and sexing. Okay, for the last keynote speaker for today's session, we will have Dr. Hendra Gunoswoyo from School of Pharmacy and Biomedical Sciences, Curtin University, Perth, Australia, with the title of his talk is Molecular Probes for Tuberculosis. So for the committee, can you please display Dr. Hendra CV? So Dr. Hendra is actually an old friend of mine. So this is his short CV. He's from School of Pharmacy and Biomedical Sciences, Curtin University, Perth, Australia. So from his additional backgrounds, he did his bachelor and PhD in the School of Chemistry, University of Sydney, Australia, and then he continued to do his postdoc in University of Illinois at Chicago, USA. So for the job experience, he became a lecturer until 2017, and then 2017 until now, he's now senior lecturer in Curtin University. So he also has many publications here. Here is just some of his awards, and also his publication this is in his corpus. Its index is 15, and then he obtained a lot of awards. Maybe it is quite small here, but I'm sure that Dr. Hendra is excellent speaker for our keynote speaker today. So for Dr. Hendra, are you here now? I'm just checking.
Yes, I am. Yes.
Okay. So for Dr. Hendra,
Share my screen.
please.
Yes, please.
Yes.
And let me share my screen.
Yeah. Okay. Yeah, we can see it clearly.
Is that okay? Yes. Can you see my camera as well?
Yes, it's clear.
Wonderful. All right. Okay. Thank you, Anna. Thank you again. And I'd like to thank all the organizing committee for kindly organizing the ISSMART 2021. I'd like to extend my thank you to the Vice Rector, Professor Alani, as well as the ISSMART Chair, Professor Faccia as well, and of course, my old friend Anna, for making all this happening. Now, I am aware we are running a bit slightly late, so I'll try to condense the message. I hope I can give you a good impression of what my young energetic lab has been trying to do in the last, I don't know, 6-7 years since I started at Curtin. So today I wanted to talk about molecular probes for tuberculosis. I'll make it very general. And oh, by the way, I'm doing this live. This is not recorded. This is my office at Curtin. So feel free to pause me, to check me if there is any questions. I'll be happy to answer them. So as Anna was saying, so this topic on molecular probes for tuberculosis, I've started this since I started at Curtin. This has become my pet project. So I'll tell you a bit about how the project sort of started. From my postdoc involvement with Alan Kozikowski and Bill Bashai at Chicago, and how that evolved into a project that I brought back to Australia, and how that evolves since then, involving the most recent work from my PhD students. So as a bit of a background, I think Indonesia is not new to tuberculosis. This is the global TB map in 2019, and obviously the bigger the circles in green, it shows you how prevalent TB is. The most recent figures in the last 5, 6, 7 years is about 10 million incident cases, and that's unfortunately accompanied with about 1.5 million deaths. Until COVID-19 hits, tuberculosis was actually the #1 cause of death in infectious diseases globally. Obviously, we all know the damage has been done by COVID is absolutely terrible, and obviously we're still in the middle of vaccination. But much of what we're learning about how COVID-19 is being handled, is being managed, a lot of these infectious disease topic has actually been done before in the past, but obviously COVID-19 is in much bigger scale. We've seen Professor Rinu's talk previously on the ACE2 and the TRPMSS2, obviously very fascinating work. So, but what I wanted to say about TB, going back, if we actually talk about the disease itself, it is caused by the infection of this mycobacterium, right? And this is Mycobacterium tuberculosis. And when you actually look at the disease manifestation, very often if you go for a visa applications, if you wanted to visit, say, Australia, definitely when I started my journey as a PhD, as a student, I had to do this lung X-ray just to make sure that, hey, you know, are you healthy, right? And so this is a typical chest X-ray of a patient that is unfortunately infected with TB, and this is what we call the presence of the lung granuloma, which is a hallmark of pulmonary TB. I'll talk shortly about granuloma very soon, but also I also wanted to mention that it has been predicted that TB, or to be more exact, the Mycobacterium tuberculosis, can actually stay dormant in your body without actually causing some damaging response. So the idea is that the latent TB, the mycobacterium is contained nicely inside your immune system, and when your immune system is obviously compromised, that's when the balance has been tipped, and unfortunately the bacteria will take over. There has been a lot of different initiatives, so Gates Foundation and other ventures, as well as this perhaps the most famous Stop TB. So India was quite I guess ambitious in saying that they will try to stop TB by 2025. Most other countries has been trying to say 2035 to 2050, but it is always good to see the government commitment to stopping TB. Now, this is what I said about the granuloma. So just to complicate what really happens in TB patients. So if you imagine that this is patient X, and this patient obviously has the mycobacteria residing within the lungs, as this person sneezes, coughs, that provides an aerosolic transmission, not very much different to what we're seeing with how COVID is rummaging around the countries at the moment. So once that gets entry into the new host, what happens is that there is going to be multiplication, and as always, when there is a new foreign body coming into your system, your immune system is going to kick in, right? So your macrophages is going to come over and it's going to start its trafficking, and obviously that has been to make the story short, has been utilized by the mycobacterium itself in order to trick the immune system so that it can expand itself in these innate granulomas. And as that granuloma matures, you will have this essentially a package or a storm of the cytokines, macrophages, lymphocytes, all the immune cells that's trying to contain the mycobacterium inside, right? So you don't want the mycobacterium to actually burst open and actually start causing the disease manifestation. And as I said before, unfortunately, so fortunately for the 90% of people who got a granuloma, as long as that immune system is in check, they'll be fine mostly throughout their lives, but the prediction is that about 10% of those unfortunate individuals unfortunately will have the immune system compromised at some point in their life. For example, in the endemic areas such as South Africa, where I spent some time there, it's not uncommon that TB and HIV actually coexist. And when that happens, obviously, then the granuloma sort of balance is broken. And now that mycobacterium is now able to just break open from this containment, and obviously that results into the manifestation of the disease. That obviously results in a cycle transmission. Now, I wanted to say that TB is obviously a really, really ancient old disease. TB is curable. We do know that. Most of these drugs that we know for the first-line anti-TB drug, from isoniazid, pyrazinamide, ethambutol, and rifampicin, all these guys were developed in 1950s. But obviously, as we all know about mycobacterium resistance and also general bacterial antimicrobial resistance all over the world, TB is not an exception. Its resistant form of MTB has been seen all across the world. anywhere from multi-drug resistance or all the way to the extensively drug resistance, all the way to the totally drug resistant TB. So it is actually quite disheartening to see that these first-line anti-TB drug that has been used for the last like 70 years, there is not newer anti-TB drugs up until 2012, 2013, where FDA and EMA approved these two new agents, bedaquiline, which is an ATP synthase inhibitor, but unfortunately accompanied with an FDA black box warning because it actually affects your QT prolongation in patients affected with MTB. So this really is put as sort of a last option, but they're now exploring a combination of this drug in other clinical trials. And the second one is this natural imidazole derivatives, delamanid. And these two agents have been somewhat recently introduced into the arsenal of anti-TB drugs. Obviously, in this realm, novel drugs with novel mechanism of actions are always needed, and this is no exception as well. New TB drugs, for those who are interested in the TB, new TB drug is an amazing sort of your go-to source for you to look at what's been happening around the world in terms of the anti-tubercular drug discovery. And this is pretty much where my story is going to start. Saying all the way, if we look at the spectrum of drug discovery from the preclinical in the initial lead optimization stage, where medicinal chemists like myself, either in academia or drug companies, that we're trying to make discoveries on the new lead optimization, and that followed by preclinical development before going to the GMP GLP talks, before obviously moving on to your human studies. And today, I wanted to talk about just two at the moment, two main targets that my group has been working on, and that is the MMPL3, which is a transporter molecule specific for TB, as well as this PKS13 enzyme, again, also present in MTB. We do start work on the ATP synthase, as I mentioned, but I don't think I'll have time to actually talk about this in this opportunity. So I'll go straight to what I think is the most sort of visual description of what we're trying to do in this field. Seeing is believing, and on the left here is the figure of the cross-section of what we call a drug-susceptible TB bacilli. So this is the Mycobacterium tuberculosis that hasn't quite developed that resistance, right? So if you like these guys on the left, you treat it with a first-line anti-TB drug and you'll kill them, right? Not a problem. But pay attention to the thickness of the cell wall, and compare it to the right-hand side here. I know that the zoom in is not exactly 1-to-1, but hopefully you can tell that there is a bit thicker cell wall in that MDR-TB bacilli on your right-hand side. And that thickness difference of the cell wall is significant. And that actually leads to the main sort of resistant mechanism, how the MTB can actually evolve over time in the last 4,000 years. And there's unique ability to stay dormant in your system, and how it can be contained all over those years without causing any damage. So my PhD student here, Shayna, so she's finishing. She has been instrumental in my lab in pulling together the bits and pieces, the projects that we have in developing potential new agents for anti-TB drugs. And so the first sort of starting point that we look at is the ability of the bacteria to make or to biosynthesize glycolic acids. So look, I'm not going to talk about the biochemistry of this, but I just wanted to say that if you go from the bottom to the up, you can see here that from the cytoplasmic side, where TMM, or Trehalose monomycolate, this guy is being synthesized through various processes from phase 1 and phase 2, involving that very PKS13 enzyme that we just talked about. So synthesis of TMM happens in the cytoplasm, accompanied or mainly instrumental by this PKS13. Following that TMM, which serves as a building block for the further cell wall synthesis, is being transported from the cytoplasm to the inner membranes by this transporter molecule called MMPL3. right? And once the TMM is in this area, then it can act as a further building blocks to make that thicker cell wall that we just talked about. So from that simplistic cartoon, it is quite obvious that from our very naive hypothesis that perhaps if we inhibit PKS13, so inhibit synthesis, and then inhibit this transport processes, perhaps we can actually compromise the biosynthesis of macrolic acids, and perhaps that will damage the cell wall, and perhaps we can start killing it better. Right. So that's pretty much what we were trying to say. So in the MMPL3, at the beginning, I'll just walk you through it, how we actually come up with these, with the initial small molecules where us medicinal chemists can actually work on. So we started from high throughput screening. So we went through about 7,000 compounds from ASDI libraries, and we did a typical phenotypic screen against the drug sensitive MTB. And from that, we found about 5 lead molecules, various publications, but the one that really catch our eyes at the time was the indole 2 carboxamide, right? So this is has been our baby since I started my my tenure at Curtin. And what I'm going to say is that, look, it possesses a unique structural motifs. It has a decent IC50 values as done by the MABA value by the MABA experiments. It has a decent cytal concentration. It is reasonably safe to healthy cells. So we use viral cells for our control. And the lipophilicity is quite high, but perhaps we can actually do something about that. So as a medicinal chemist, look, I'm not going to try to bother you with too much of a synthetic schemes, but I try to show you that this is basically what we do. So we create individual structural modifications to different parts of the molecules, and then we take that into biology, and that gives us a feedback as to which structural modification would actually work best in terms of functional efficacy, but also vice versa, which substitution or which replacement, which extension actually did not do well for our target. So we've made about 200 analogs. That number is growing by now. In my own lab, we probably have about another extra 200. But from there, we're trying to build this body of structure activity relationship. And eventually, hopefully, we can then keep optimizing potency, making sure it's more drug-like, and more importantly, it has good activity in vitro and obviously in vivo. This is a very quick rundown through MABA. So it's obviously an assay that relies on the presence of resozurin. And the idea of the assay is that if your mycobacterium cells is alive, is functioning, then you have this reducing environment. And what it does is that the mycobacterium is going to reduce this resozurin into resorufin. And this guy is going to be strongly fluorescent. And you can do so upon mixing of your testing compound as well as the bacterial suspension with a bit of the dye, the alamar blue, then you can start seeing this kind of data. And this work is done with my lifelong collaborators, Dr. Sejun Lun and Professor Bill Bashai, and this work is done at Johns Hopkins University. So quite initially, this is now 2013 now. There is follow-up work on 2016 and 2018. But effectively, what we're doing as a drug discovery group here in Australia, we make those molecules, we identify them, we purify them, we HPLC them, we then send it over to our colleagues. And practically, once you go through those screening processes, you will then select 1 or 2 hit molecules that you will then put in mice to look at bioavailability. So as one of our sort of prototypical compound, we did indole 2 carboxamide 12. And as you can see here, the MIC to the drug sensitive strain of MTB is very reasonable at 13 nanomolars. Viral cell IC50 is over 50 macromolar. So if you divide these 2 numbers, you'll get a selectivity index of which is very respectable, more than 4,000. That gives you a nice safety window. And more importantly, it is orally bioactive as well as it also has multiple activities against the resistant forms of MTB. So these are typically what we do. And also just to give you a bit more flavor as to what we also do, once we find these compounds that is active in vivo, we typically follow that up with the generation of resistant mutants in the lab. And following that, we sequence, we perform a whole genome sequencing, not only to the mutated bacteria, but also comparing it to the naive mycobacteria. So that way you can see what sort of single nucleotide polymorphism that has been occurring in the mutant species. And that gives you some insights into the possible mechanisms of how these compounds work. So all this work has been published previously, and this is a cartoonic representation of the MMPL3 as a transporter molecules. And these 6 different numbers essentially tells us the different molecular probes that has been shown to be acting as an anti-TB compound that has been shown to inhibit MMPL3 as that target. So just to show you that that indole 2-carboxamide us is this compound 6, so in red. So that's the site of the SNP, but also other compounds like the adenosine and also the BM212 by the Italians. They also they target different parts of the MMPL3, but they all ultimately come down to the inhibition of MMPL3. So this is the kind of work that we do to show that our compounds work via MMPL3. Now, combining all those together, so what happens to the MTB when we actually treat them with one of our best compounds? So in this case, we took inolamide 1, we published this about 2 years ago. So if you look on the figure A, this is untreated, the bacteria is quite happy. And this is bacteria upon treatment with 10 times MIC of the first line anti-TB drug with isoniazid. So you can start seeing these compromises in the cell wall as expected. That's how isoniazid works. And again, if you treat it with our compounds at 10 times MIC, not only you start seeing some compromises in the cell wall, you start seeing these blobs and these openings, and eventually you can see you can start seeing some cells rupturing. So this goes hand in hand with our genomic studies, with our electron microscopy studies, as well as our PK/PD knowledge. So where to from there? So in here, again, we thought, well, PKS13 works, MMPL13 works, MMPL3 works. So when we started the project in my group, we, again, being medicinal chemists, we worked with this library of compounds. And just to show you that we actually found a hit, HCC118, and that was a initial molecule. And obviously, since then, in the next 2 or 3 years, we've been developing the series. I don't think I have time to actually talk about all the chemistry, so I'm sorry for the organic chemists out there. This is all published, but briefly, you can make these compounds from these readily available acetoacetates, condense it with benzoquinones under copper catalyst condition. You form this ester, benzofuran ester, and then you can hydrolyze it, and then you can do a manic type reaction or to install this piperidine, and you can do many more structural modifications. You can change it to an amide. You can cyclize this, as you will see, and obviously for the most interesting compounds, we can do X-ray crystals as well. So I will skip through all these just to give you the idea that we made these compounds, right? We made these compounds, we NMR'd them, we analyzed them, we purified them, and so being a medicinal chemist, obviously, we look at the MIC values. So as you can see here, we're changing one structure at a time, and how that actually brings about the changes into our profile. So obviously, through generation of many analogs, we can actually get some decent compounds. And obviously, those most decent compounds were the one that's going to be targeted further for more advanced in vitro studies and perhaps in vivo. So again, this is a very similar figure as you saw, the serum inhibition titration studies. So as you can see here, these compounds here, the Coumastan 2111, it has still a decent activity up to 1 to 32 dilution. Yes, the dose could be a bit high, was a bit high, but at least in principle, we can actually say that MMP-L3 inhibition does bring about potent anti-tubercular activities in vivo. From there, as most sort of antibacterial drug discovery group, we do various cytotoxicity. This one is to viral cells. We also do typical cytotoxicities to other more benign cells, just to show that our compounds are not randomly killing cells, right? Again, those works are followed by a series of target identifications. So we use genomics a lot for our best compounds. Again, examples of the SNPs, and this is consistent with either both PKS13 inhibitors targeting PKS13 and MMP-L3 inhibitors targeting MMP-L3. Right. So at this stage, we've confirmed that PKS13 is a viable target. We're not the only group doing this. The other people in the US, James Saccatini work has also been doing this work, and we actually sent we've been collaborating with them in the fact that they've been sending us some plasmids for us to be able to grow them, to express the PKS13 enzymes, and so that allows us to actually look at in vitro evaluation of our newer analogs. So all this work is still in progress, and obviously for the structure-based drug design people, they actually have the crystal structure of the PKS13, so that helps further in designing new molecules. So since then, look, I know I'm running out of time. Since then, I just wanted to give you some more recent updates. So these are a series of newer papers. So earlier this year, again, hopefully you remember some of these structures. So these Coumastans, we did more typical, I guess, AAC style antimicrobial agent chemotherapy style paper, where you just take some of your best molecules and then you do a bunch of the classical in vitro, in vivo, and PK/PD. So this pretty much I'm going to show you exactly that. So as you can see here, so different molecules have different MICs, and it also shows you the comparison to isoniazid and rifampicin, and this shows you the different mutation, and these are obviously a mutation that has been designated in the PKS13 gene. Once we establish all that, what do we do? Sorry for the small figure. We also did this, what we call a kill kinetics assays, where we are just following over time the ability of our compounds at various concentrations to be able to reduce the population of the CFUs in our pool. Following that, we also did, as I said, a bit of PK, quite common. You cannot really advance good compounds as potential drugs if your PK is obviously compromised. So what we can see here, the T-half, so the half-life is decent at about 5 hours. The Tmax and the Cmax is reasonable, by no means perfect, but this bioavailability of 20% is definitely acceptable as an initial starting point. And I know that this conference is all about smart molecules and how we can connect compounds to natural molecules, to public health, to cancer molecules, all the way to the sperm modifying agents, perhaps, as the previous speaker has alluded to. I just wanted to say that this sort of coumastrol species, they're actually present naturally in high levels in clovers and chickpeas. I mean, I know they're not exactly the same, but again, this shows you that there are many scaffolds, either from natural products or synthetically made, that are just waiting for them to be discovered to be used for different uses, right? So in this case, we managed to make a good use of the Coumastans in mycobacteria. So where to from here? My lab is also focused on looking at very, I guess, mainstream epigenetic controls of mycolic acid biosynthesis. So we've been looking, again, without going into too much detail, we look specifically at the mycolic acid biosynthesis, and we are looking at the epigenetic phosphorylation, dephosphorylation that is obviously nicely orchestrated by the MTB species all the way to our macrophages to trick our immune cells into making them dormant and into making them surviving for a very long time. So again, the idea here is that, again, without going into too much detail, this is published work. We are hoping to look at, can we perhaps repurpose kinase inhibitors for TB? Right. So this work is ongoing. I wish to update you in the next few years. But our most recent papers on the MMPL3 has resulted in a couple more publications this year. So again, just to show you a variation of what we did. So we look at bioassociative replacements of the indole ring into quinolones, into naphthalines, and other agents. We also looked at other molecules to incorporate different type of adamantane-containing groups. In this case, we're using adamantanols, which we've shown to improve water solubility. And very lastly is our most recent paper to show that the indole 2-carboxylates can actually be used, and shall I say, can be switched in terms of their activities from killing mycobacteria to killing pediatric brain tumor cells. This has been published, and there's actually a second paper just recently accepted as well. But this is pretty much what we're trying to achieve in the lab now. So how can we taste apart the anti-tumor activities from the antibacterial activities, and how do we actually make the link between those two? So we've been currently looking at the mechanism of actions, and obviously this is an ongoing project. All right, so sorry I went over time, but here is my acknowledgement slide. I cannot thank all my group members at Curtin. I try to highlight their work as much as I can. They are a bunch of young, energetic scientists, and I'm sure most of the audience here are too. So yes, that's all from me. Thank you very much.
Thank you very much for Dr. Hendra for delivering his interesting talk about TB and drugs for TB. Okay, ladies and gentlemen, now we are moving on the discussion session. So our committee has already listed all the questions from the attendants. I would like to ask Emma to share screen the question for our attendance today. So for Professor Enwatwa, Professor Mariana Ketudak-Keynes, and Dr. Hendra Kunaswoyo, we will have questions to you. Please, maybe this is gonna take a few seconds for the screen to come up. Oh, Hendra, I forgot to ask. Can you stop screen? So
I will. Sorry about that. My apologies. Sorry.
Okay, so for the first keynote speaker, Professor Enwatwa.
Yes, I'm here.
Okay, thank you so much. So we have several questions for you.
Sure, I can see that.
Okay,
the first question is where and what kind of mechanism the bioactive compound of ashwagandha work inside cell or tissue. Well, as I presented, there are various bioactive components. Each of the component has a different mechanism of action. For example, the anti-cancer activity will take place by some mechanisms, and there are multiple of them which we have already demonstrated using cell culture system or even the animal system. So it depends on what component we are talking about. So as a nutshell, I like to say that it is multifunctional and multiple mechanisms of action.
Okay.
Okay. The second question is, according to NOFAR on mice model of cancer, how was the cage environment adjustment for their daily care? Yeah, it's the standard protocol. We keep them on the day and night cycles of light, and we have their feeding in the cage. So it's a standard protocol.
So the same as
it's called the nude mice. Yeah.
Yes, thank you. Okay, so we move on the next slide for the next questions. Next question, please.
Okay, I can see that one question for me. Is there a receptor which make the ashwagandha become selectively to cancer cells? Well, we have not completely dissected the mechanism for anti-cancer activity, but from the ones we have done so far, it seems like it targets the molecules which are enriched in cancer cells. Right? Yeah. So next question to me again is, which one from the compounds of ashwagandha that has the lowest binding affinity? Is it described the true result in experimental research? I don't get this question. Affinity to what? So you're asking me the lowest affinity. Is it regarding the cancer, or I don't know what activity you're talking about? So, of course, as a general, what we have seen by computational analysis and the experimental data, the different vidanolides that they have different binding ability to the receptors that we have studied so far. For example, I was showing you the data on the cell surface receptors that are involved in coronavirus infection. So with the different vidanolides, like vidanolide A, B, vidanoside 4, 5, they have the different binding affinity.
Yes, thank you. Of course, for the next one. Still for...
No, that's not for me.
Okay.
Okay.
Thank you.
That's for Mariana.
For Marina, yes.
Okay, so for actually at this moment, we still don't have any baby cattle born yet, because cow takes 9 months for pregnancy.
Like human.
Yeah, so we actually next month we are hoping for about 200 baby calf born. Hopefully it'll be more than 80% female. And so actually this technology has been done, the sex sperm things has been done for the last 20 years using flow cytometer to separate the X and Y sperm, and the baby born are normal. And actually for our work, using the antibody to separate the X and Y sperm, so we just destroy the Y sperm, and the X sperm, the motility, the morphology are normal, and using that for in vitro fertilization, the development of the embryo are all normal.
Okay, thank you. For the next questions.
Yeah, can this be applied to other animals? A lot of people been asking me about it. The pig farmers want me to do their work on the pig sperm also. However, actually there's more basic science that we would like to do. What does our antibody bind to? Which molecule on the head of the sperm of the cattle? So to answer this question, I think the molecule of the antibody recognized will be different from each animal. So I don't think our antibody would work for goat and buffalo or cats or dogs.
Yeah, okay, thank you. But you chose big cattle like bovine. So maybe it can be applied to smaller cattle like goat maybe?
We have not tested on goats yet, but we did test on, so we started with dairy cattle, and we tested it on some beef cattle. It's still the same species, but just want to see if there's any difference or not. But it worked okay, worked well on different breed of cows also.
Okay, yes, thank you very much for your answers. So for the next one, we have questions for the last keynote speaker, Dr. Hendra.
Right. It is my turn now. Thank you, Anna. From Andriana Kusuma Pertiwi. To Dr. Hendra. Please just call me Hendra. For TB drugs, can they be used for COVID-19? Thank you. It's excellent question. Now, we are, short answer to your question is at the moment, no. Right. So firstly, it's COVID-19 is a virus, right? It is a SARS-CoV-19 virus, SARS-CoV-2 virus, sorry. And what we're learning about the mycobacterium is obviously is that the bacteria and the virus share very, very different structural proteins. But obviously the mechanisms, the knowledge that we gain from how the mycobacteria can invade and invade the cells and then evade our whole immune system can obviously be translated into how perhaps we can design future strategies to tackle COVID-19. I guess the mutation that we are seeing at the moment around the world with the Delta, obviously, Epsilon is probably not cursing us, but probably going to come sometime soonish rather than later. And obviously, this forces the whole pharma companies to make sure that they can keep up with the demands to make sure that the vaccine boosters, from various companies are making sure that they are addressing these sort of seasonal variations of the COVID-19 strain. So short answer to your question is, as a treatment agent, no, TB drug cannot be used for COVID-19. We have seen examples of, obviously, and some of the media has been really twisting this, but obviously you guys heard about the hydroxychloroquine, the anti-malarial drugs, and how that actually they say that it could be potent for COVID-19. Now, there has been a lot of articles as such, but really, I think there is a lot of gray literature out there too. So we need to be extremely careful when we're actually saying that, this agent should be used for COVID-19. Obviously, everyone here in this audience have heard about how ivermectins are potentially an agent. Again, all these type of the work, although they have been built on the premise, on the fact that they are known drugs, but still evidence are still missing for those COVID-19 use. So I hope that answers your questions, Adriana. The next one, Dr. Nia Kurnianingsih. How is TB in Australia? I can tell you almost non-existent. So we've been fortunate. So as I said before, TB, I think hit the most probably around 1900s. So pretty much when the anti-TB drugs were discovered or were, actually released in the 50s, I think at that time, Australia has probably gone down to like 20 cases per 100,000 people. And I'm pretty sure in the last 3 or 4 decades, that number has gone down to like 2 or 3 per 100,000 people. So hardly any. We can say that TB has been completely eradicated in Australia. But by TB, I meant the active TB, all right? Not the latent TB. The latent TB is still roaming around, possibly in every single one of us at the moment. At least the prediction is that it is in up to 1 in 4 populations. So I answer the prevalence, and are there any different clinical manifestation with TB in Indonesia? So again, not relevant to the TB in Australia, but what we've done with our collaborators in Johns Hopkins was that I actually spent some time in South Africa during my postdoc. And so I was fortunate enough to stay in Durban, South Africa, and we actually see live how the patients actually give their sputum, their air, right? And then how we collect them from the patients. We obviously grow them in the cells in a lab, and then we send that over to the Johns Hopkins. So our collaborators in Hopkins has quite a massive collections of all these sample patients from South Africa. So all the way from the multi-drug resistance to the extensively drug-resistant TB. So I hope that gives you the kind of scope of studies that we do. So yes, not so much to Australian one. We haven't done the Indonesian one, or at least not with my group of collaborators. But obviously, many people around the countries, probably around Thailand, have also done very similar studies.
Thank you. Do we still have question for Dr. Hendra? Okay, so if you don't have TB in Australia and you got samples from overseas, from outside Australia, it's not a problem for the border security?
Oh, it is a big problem. Australia is you guys might have heard about this. We are quite strict in terms of our biosecurity laws. I mean, look, let's give you an example. I mean, banana, right? I mean, you cannot just carry banana inside Australia. I'm sure you guys heard about this. But yeah, I mean, it's TB is definitely not prevalent in Australia for sure, and I mean it is a good thing. For example, if you also see malaria, right? Malaria, if you look at the most recent news in China, they actually claim that the Jinghaosu, the artemisinin, have actually been really effectively killing malaria in China. So we can see how malaria was a big issue in one country, and then they can completely eradicate that. That would be something that we hope that we can achieve in those endemic countries moving forward.
Okay, so we have two more questions for you, for Dr. Hendra in the
Okay,
chat.
okay, right.
So, first one,
Thank you for the
to
informative.
synthesize all the molecules, do you use special tools or manually? And then the cell viability assay is the mycobacterium added in the ferrous cell.
Right.
And after that, you do the assay for the selectivity and the effectiveness.
Got it. So I'll answer the first one for Maratu Soleha. So do you synthesize the molecules specially or manually? We use organic synthesis. So we use organic synthesis as a method to make those compounds and manipulate those specific functional group transformations. So all the way from your alkane, alkene, alkyne, alcohol, carboxylic acid, all that. We know that there are specific chemistry that can be applied to those functional groups. And once we make them in a lab, we need to purify them by a series of column chromatography. And once it's done, then we look at the nuclear magnetic resonance spectra of the proton and carbon. And we typically then go to HPLC to purify them. We're hoping to see that one peak nicely. And only then we'd actually send them for testing to our collaborator. So short answer to your question is a mixture of tools, but standard tools in organic synthesis for drug discovery. And the second part of the question is, for the cell viability assay, I'm assuming you're talking about the microplate Alamar Blue assay. Is the mycobacterium added in the viral cell? And after that, you do the assay for the selectivity and the effectiveness. I get what you mean. So the MABA is the platform, right? So firstly, what we do is once you make the compounds, you make sure that it's pure, then you apply. So you make a broth of your mycobacteria, and then you add increasing concentrations of your test compound, right? So at a lower concentration, obviously, it's not going to kill the bacteria, but at a higher concentration, then you start seeing that growth inhibition. And it is that spectrum of colors that will allow you to get that middle point that we call the MICs. And so that's how we do it in the first instance. Now, once we know that the compound is toxic to the mycobacteria, then we repeat the same assay, but instead of the mycobacteria, we take the viral cells and then your compound in here. And again, you do that spectrum of concentration increases. And again, you take the middle value, the MICs, and then you compare the MICs from the mycobacteria to the viral cells. And effectively what you want is a higher selectivity for the mycobacteria compared to the viral cell. So that way, if you have a good selectivity windows, that will give you a higher confidence in giving you that nice safety therapeutic window. I hope that answers your question.
Yes, thank you so much for Dr. Hendra. Oh, I'm sorry, there is one more question for Prof. Renu in the last chat. Is Prof. Renu?
Yes, I'm here.
Still here?
Sure.
Okay, thank you. Prof. Renu, there is one more question for you.
Sure.
What method do you use to enhance the certain metabolite in ashwagandha plant?
We make different kind of extracts. So basically, there are extraction protocols which can give you one kind of bioactive better than others. And then we are also making, specific plants which can be enriched with a specific component.
Okay, thank you very much for your answer. So for everyone, ladies and gentlemen, I think this is the last session for the plenary session for this morning. I would like to thank to all our keynote speakers, Professor Renu Latwa, Associate Professor Mariana Ketudet-Keynes, and Dr. Hendra Gunawarsowoyo. Please give big applause to our keynote speakers today.
Thank you. Thank you, everyone, for having us.
Thank you very much.
The next session, I would like to ask for MC Yora to guide the presentation or to guide the event for today.
Okay, thank you so much, Ana Safitri, as moderator for plenary speech in this morning. And then for today's documentations, I would like to kindly ask to all ISSMART attendants to open their video. We will take photos for ISSMART first day. Okay, the committee will take some documentation screen by screen. Yes. For the first screen, 3, 2, 1. Yes. Second screen, please to open your video. Yes.
You mean by open the camera, no?
Yeah, open the camera. You're right. Thank you. Okay, 3, 2, 1. Okay, and last screen. 3, 2, 1. Okay, thank you so much for all ISSMART attendants.
Okay.
So I have a question about the 3 session. How do we get into the session? Is there any breakout room to join or something?
To all our keynote speakers, I would like to award our certificate of gratitude. Please, committee to share screen the certificate for keynote speakers for today. We will also send the certificate to our keynote speakers email. So first, we would like to thank to Professor Renu Atwa, PhD, as contributed as keynote speaker for ISSMART 2021. So for the next one, we would like to express our gratitude to Associate Professor Mariana Ketudat Keynes, PhD, as our keynote speaker for ISSMART 2021. And for the third keynote speaker, we would like to thank you very much to Dr. Hendra Gunaswoyo as our keynote speaker for ISSMART 2021. Thank you very much.
Okay, thank you. Dear ISSMART 2021 attendants, if you are presenting, you can join room A or B or C according to your schedule. If you are not presenting, you can also join parallel room and please ask the meeting host for this. With the end of the event, in the main room this morning, I am Yorafika Dwi Bibangga as a master of ceremony on this. I would like to say goodbye to you all. I apologize if there are any mistake. See you tomorrow. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you. Please joining all breakout room that will already provided by the committee.
Biar aku bisa masuk room ya. Maksudnya biar bisa ganti gitu kan, yang di streaming kan biar ganti. Oh.
Analysis from Indonesian Center for Rice Research. The presenter has just changed from Dr. Yudi Sastro. And so, okay. Okay, thank you. Okay. Dr. Aris Hermansis is a PhD, has a PhD of plant science at the University of Adelaide 2014. And he's now coordinator of program and evaluation, and also is a manager of rice seed management unit of ICRR. He published more than 80 papers, and also he released many rice cultivars. His title of presentation today is Increasing Nutritional Values of Rice Through Development of Healthier Rice Varieties and Its Byproducts Utilization. Okay, Dr. Hermansis, please begin your presentation.
All right. Thank you very much, Dr. Tomohiko Sasase, as the chair. And first of all, I would like to thank the organizing committee for inviting us to this important meeting. Let me share our presentation today. All right. I hope you all could read will see our presentation. First of all, we would like also apologize that Dr. Yudi Sastro, the director of Indonesian Center for Rice Research, should be present his talk this morning, but unfortunately he has another important agenda, so he asked us to represent him in presenting or sharing some of our research achievement in Indonesian Center for Rice Research, especially on the development of healthier rice. Our talk today will be on increasing nutritional values of rice through development of healthier rice varieties and its byproducts utilization. Before I start the talk, maybe I need to introduce our institution if someone who attending this meeting not really familiar with ICRR. So we based in Sukamandi, Subang, West Java. It's about 2 hours from Jakarta. So our office is actually in the middle of the rice field. So I hope that you could visit our station or our headquarters someday. We produce or we develop many kind of rice varieties and also many rice technologies to support Indonesian rice production. All right. Related with this seminar, I think many of you already aware with the nutritional problems that faced by Indonesia. Actually, all countries in the world may be having problem on nutrition, but we aware that Indonesia not only has single burden, but we have a double burden in malnutrition. For example, we have a problem in stunting. We still in around 30% of Indonesian toddlers, and then also problem with wasting and also problem with overweight. In addition, many pregnant women in Indonesia still have problem in anemia, with the figure still very high, about 48% in 2018. We call this problem as hidden hunger because it's related with the macronutrient deficiency, like vitamin or mineral, important mineral like iron, iodium, folic acid, zinc, and vitamin A. And of course, this significantly affects on the quality of human resources in Indonesia. That's why Indonesian government really focused to overcome this problem. This is some information, maybe general information about some problem in nutrition is causing hidden hunger. Three main nutrients that now become focused by WHO and other global organizations because it's a significant impact on human health, like iron, zinc, and vitamin A deficiencies. And that's three nutrients that we now focus in our breeding program in Indonesia to develop new varieties with better content in iron, zinc, and vitamin A. We know that we need iron for brain development, immune function, and also for good energy levels and healthy pregnancy. We need zinc for growth and development, immune system.
Recording in progress.
Other health issue. We need vitamin A for vision health and preventing infection, and also fetal development. Iron is also important because it causes anemia. Zinc can cause stunting, one of a big problem in Indonesia. And vitamin A deficiency cause impaired vision, of course, and poor pregnancy outcomes. There are several strategies that we can address there are several strategies that we can use to address hidden hunger. First, of course, the most ideal strategies is by dietary diversification, by improving the dietary of human, such as from the vegetables, from fish, or from egg and others. But of course, this needs more cost because not all Indonesian people would buy, balanced diet. And also the second strategy is by using supplement, and this is one of strategies that's used by our government by giving supplement to toddlers in Indonesia. And the third is fortification. We know that many food in Indonesia is already fortified, like oil or milk or other food. And the last which we use is by using biofortification, by improving the nutrient status in important crop like rice. For you who is not really familiar with biofortification, biofortification is a process to increase the bioavailability and the concentration of nutrient in crops through both conventional plant breeding or recombinant DNA technology or genetic engineering. The process by which the nutritional quality of food crop is improved through agronomy practice. So besides using varieties, we can also by agronomy practices like fertilizing or others. And biofortification increase nutrient levels in crops during plant growth rather than through manual means during post-harvest processing of the crops. Why we use rice for biofortification? Of course, rice is the staple food in Indonesia. It's the main source of carbohydrate for more than 90% of Indonesian people. Besides containing carbohydrate, rice also contains protein, vitamin, and other micronutrients, also some bioactive substance like antioxidant, which is in maybe in normal rice or in regular rice, the content is a little bit small, but we can increase through breeding program. So rice is very potential to be used for biofortification. The benefit using rice in biofortification program, of course, the first is it will be sustainable. By one-time seed entry, we can use this for everlasting application in the targeted area, especially in the remote areas. We can just distribute the seeds, and then the farmers can grow and distribute the food or the rice to the people in that area. And it can be massive because every people could consuming the rice and get the benefit. It's efficient and practical, no need special treatment, and also it's natural because we do not need to change people custom. For example, for Indonesian people who eat rice 3 times, they still can eat rice 3 times, but with a better rice, of course. There are three main research focus in ICRR that we currently doing to improve the nutritional quality of rice. First, by the breeding program to improve the macronutrient content of rice. In this case, it's zinc, iron, and vitamin A. And in this talk, we will mainly talk about zinc because this is the most progress one. And then we also develop improved pigmented rice, including black rice and also red rice. And also we try with the processing technology, we try to utilize byproduct of healthier rice for human consumption. First, let me share about the progress of the breeding for high zinc rice in Indonesia. We use mainly conventional breeding program to improve the zinc content in rice by using crossing. We cross many rice germplasm with high zinc content, and then we select the progeny. We do yield trial before we do multi-location trial and release the new varieties for the farmers. This is one of the new variety that contain high zinc in rice. We call in Pari Nutri-Zinc. It released about 2 years ago in 2019 as a collaboration between ICRR and Indonesian and International Rice Research Institute in Manila, Philippines. The average zinc content of this rice variety is about 29.4 ppm. With this figure, it can provide at least 1/3 of zinc requirement for average requirement for zinc for Indonesian people. And it has potential zinc content up to 34 ppm. And then it has soft texture. So because most of Indonesian people like soft or pullen texture, and the shape also is slender. It's similar to general Indonesian people preference. It has quite a good yield. It has a 9 tons per hectare average yield, comparable to popular variety like Ciherang, and it has quite high yield potential up to 9 tons per hectare. The duration also similar to popular variety like Ciherang, about 115 days. And it can be cultivated with normal cultivation technique according to integrated crop management practices. Another variety that we released last year is Inpago 1340s. If Inpari Nutri-Zinc is targeted for lowland area, Inpago 1340s is targeted for dryland or upland area. It has potential zinc content up to 34 ppm in the brown rice, and it has quite high also in protein content. It's about 9.8% with the average yields of 6.5. It's also resistant to blast disease, which is important disease in upland, tolerance to aluminum toxicity, and also tolerant to drought stress. Besides the improvement in macronutrient, we also conducting breeding to improve pigmented rice in Indonesia. We know that Indonesia has very diverse rice germplasm with a diverse color of aileron. Pigmented rice contain important substance which is good for health, like anthocyanins, proanthocyanidins, phenolic acid, flavonoids, tocopherols, and phytic acid. Unfortunately, most of farmers in Indonesia currently still grow traditional rice varieties to produce black rice or red rice. Of course, these traditional varieties has low yield, and also it has long duration, sometimes up to 5 months. Farmers can harvest their rice. Pigmented rice has potential to lower the risk of chronic disease like diabetes, cancer, and cardiovascular, of course. And we need improved pigmented rice to produce higher yield and more resistant to environmental stresses to help our farmers who are eager to grow this healthier rice. There are some new varieties that already developed by ICRR with a pigmented aileron. We have, we call Palmera, this is red rice. And then we also has Pamalan, this is also red rice. And the last is Jelitang, which also it has black aileron. I give one example for Jelitang here. Jelitang is black rice which have a black aileron. It developed by crossing Ketan Hitam and Pandan Wangi Cianjur. So this is we make using a conventional breeding program, and of course compared to the local varieties or traditional varieties, it has shorter duration. Farmers can harvest this rice variety only in 13 days. And then it has quite a good yield, about 6 tons per hectare compared to traditional varieties, which usually less than 4 tons per hectare. And also it has cooked rice texture. This is important because when many people not really want to eat brown rice or black rice because it has a hard texture, but by developing new varieties with low amylose content, we can get like a black rice with a soft texture. So people can accept more easily to get to eat this rice because it's softer than general black rice. And last year we also just released in Pari Arumba. This is black rice, and beside it's red rice, and beside red it also has aroma. It's aromatic rice, so it's good with a good flavor. We hope that this rice varieties not only increase the health but also increase the adoption. It has quite good yield potential and yield average, and the phenolic content also quite high. It has soft texture and resistant to some of rice disease. This is some data for the phenolic content, anthocyanin total, and phytic acid and of pigmented rice that we developed for the older variety like in Pari 24, in Paha 7, in Pago 7, and IFC Bundong. Its phenolic content quite diverse. The highest is in Pari 24 and in Pago 7. For the anthocyanin also is related with the phenolic content. And interestingly, for the phytic acid, we have in Pago 7, which quite high in phytic acid. And I think this is our last slide. We not just develop new varieties. We try to utilize byproduct of rice. Usually, we do not use like broken rice or bran rice for human consumption in Indonesia. Usually, we just use this for husbandry, but we try to improve the utilization of this actually what we call important product actually. So we try to develop technology to make rice milk with fortified rice milk. So we use broken rice or pigmented rice and high zinc varieties for milk. The nutrient content is quite high, 599 microgram of folic acid and 600 microgram of riboflavin. It has antioxidant activities 700% higher than goat milk. Of course, it's cholesterol free and lactose free. This product is already registered for the patent and now in the progress for the commercialization. As a summary, rice as the staple food has a very important role in determining dietary pattern of Indonesian people. Increasing the nutritional value of rice is needed to supply essential nutrients in their daily food. And then improvement of the nutrient in rice can be achieved through development of healthier rice varieties, such as biofortified rice or high zinc rice or antioxidant-rich pigmented rice. And also we can use the byproduct for healthy foods such as rice milk, which could benefit for human health and increase the economic value of rice. I think that's all for us, and we hope we can have a discussion later, and we really welcome for suggestion or feedback from the participant. Thank you very much.
Hey, Dr. Helmansis, thank you for your informative presentation. I think it's a very valuable project in Indonesia. And now let's open question and comments from the audience. You can ask question by chat, or also you can turn on microphone and ask to Dr. Helmansis. Any questions? No question.
I'm not sure if participants here are familiar with the breeding or with rice improvement program.
Okay, may I ask first question, Dr. Helmansis? I think your talk is very important and it's a very valuable project. And I understand you are improving and developing a land races of Indonesian rice.
Yeah.
And I think the problem is common among the Southeast Asian countries. So I think many other countries may also developing such kind of improving or developing rice because they eat rice as their staple food. So my question is there any advantage of your project compared to other countries? Or other country also developing such kind of rices?
All right. Of course, this kind of project is common in Indonesia, in ASEAN, the Asian countries, of course. But we have each country has specific target environment and also a specific problem like pest and disease. That's why we need a specific program for Indonesia. For example, pest and disease in like Vietnam or in Japan maybe will be different with Indonesia. And also the environment also different. So we need like a specific rice varieties which could adapt specifically in Indonesia, is resistant to specific pest and disease in Indonesia, as more resistant to environment condition in Indonesia, and more importantly has Indonesian people also has specific preference in rice in grain quality, and which of course different to other countries. That's why we need a specific breeding program for Indonesia.
Okay, thank you very much. Any other questions from audience?
No.
Okay, thank you very much, Dr. Helmansis.
Thank you.
Okay, let's move on oral presentation session. In this session, each presentation is 10 minutes, and we make 10 minutes discussion time after every 3 presentations. Okay, I'd like to welcome our first speaker from Kyoto University, Japan, Dr. Tatsuya Maekawa. His title of presentation is Brain Abnormalities in Spontaneously Diabetic Torii, SDT Fatty Rats. Dr. Maekawa, are you ready to start your presentation?
Thank you very much for the introduction. Can you share my presentations on the, can you hear me?
Yes, it's okay.
Thank you very much. At first, thank you very much for accepting my presentations and giving me this opportunity. My name is Tatsuya Maekawa. I'd like to make a presentation on behalf of Professor Ota's laboratories at Kyoto University. My title is Brain Abnormalities in SET Fatty Rats. This is agenda. First, I'd like to talk about the diabetic dementia and current status of therapies. Next, I will talk about my study and its result. Finally, I will summarize the talk. In recent years, there has been an increasing number of reports on the relationship between the diabetes and central nervous system disease. In particular, a lot of cohort studies have reported that diabetes is a risk factor for Alzheimer's disease and other diseases. The onset of diabetes causes various disorders through mechanisms such as atherosclerosis, microvascular disease, glucotoxicity, and insulin resistance, and also causes pathological changes in the brain, such as vascular lesions and Alzheimer's disease, leading to dementia. It is becoming clear that diabetes and dementia are closely related. However, at present, effective treatment for the pathogenesis of diabetic central neuropathy has not been established. The pathogenesis of neuropathy is very complex, which is one of the reasons why it is difficult to establish a treatment. Therefore, it is important to elucidate the pathogenesis of neuropathy using experimental animals as a basic research. I will now introduce the result of our research. The purpose of this study is clarify the pathogenesis of central neuropathy in the STT fatty rats, and we examine the morphological changes and inflammation-related gene expression in the brain. We examine the cortical thickness, number of hippocampal pyramidal cells, and mRNA expression in the brain of male rats 32 and 58 weeks of age. As some of you may know, SDT fatty rats are a model animal that exhibit hyperglycemia, hyperlipidemia, and hyperinsulinemia, hypertension, and famous diabetic complications. As a result, cortical thickness was measured as shown in the left HA stained image. The results showed that there was a significant decrease in cortical thickness in SDT fatty rats at 58 weeks of age. Next, we counted the number of pyramidal cells in the CA1 and CA3 regions of the hippocampus. As shown in the figure on the left, decrease in the number of pyramidal cells in both CA1 and CA3 regions was observed in SDT fatty rats at 58 weeks of age. On the other hand, 32 weeks of age, there was no effect on the number of pyramidal cells as well as cortical thickness on the previous page. When we examine the inflammation related to gene expression in the brain, we found that S100A9, NF-kappa B, and TNF-alpha mRNA expression was increased as shown in the figure. Okay, let's sum up. In this study, various changes in the brain were observed at 58 weeks of age rather than 32 weeks of age, suggesting that morphological changes in the brain may be caused by persistent hyperglycemia. Although this may be only some of the brain change, the SAT fatty rat could be used to elucidate the pathogenesis of central neuropathy associated with diabetes and to develop therapeutic strategies. That's all I have to report. Thank you for listening.
Okay, thank you, Dr. Maekawa. Okay, next presenter is from the National Institute of Advanced Industrial Science and Technology, Japan, Dr. Dong-Yang Lee. The title of presentation is Ashwagandha Extracts for Oxidative Stress and Lipogenesis. Dr. Lee, please feel free to begin your presentation. Excuse me, could you turn on the microphone, please?
Oh, okay. Sorry, I can play this video from my computer.
Okay, thank you.
I can't hear anything from this one. Sorry, can I replay this video? Because no sound in this video. And I can share from my computer. Sorry, can I share the screen?
Yes, it's okay.
So can you stop this screen from your side? And then I will share from my computer. Sorry, I won't share from my computer. So can you stop this video and I share from my computer? Because this video have no sound. 没法控制。
Sensei Tasase, maybe for the next presenters.
Oh, okay.
Yeah.
Okay, so I'd like to start third presentation, Dr. Huayue-san. Are you okay, Dr. San?
Yes.
Okay, so the next presenter.
Yes, I'm here.
Okay, next presenter is also from National Institute of Advanced Industrial Science and Technology, Japan, Dr. Huayue-san. Her presentation title is Effect of Ashwagandha Extracts on Muscle Cell Differentiation. So Dr. San, please start your presentation.
Okay. So shall I do live presentation now? Because previously I chose to play my video. But if the video is also no sound, I can do live presentation now. Okay. Yes. Can you see my slide? Okay.
Yeah, it's okay.
Hello everyone. My name is Huayue Zhang. I'm a first-year PhD student of University of Tsukuba, Japan. And today I'd like to share my research, and the title is Effect of Ashwagandha Extract on Muscle Cell Differentiation. So let's start with the introduction of ashwagandha. Ashwagandha, also known as Indian ginseng, is an important medicinal plant that has been commonly used for its health-promoting effects for over 3,000 years. It has been demonstrated that ashwagandha extracts prepared from different parts of the plant show different ratio of active ingredients, including risaflin A and resinol. And the bioactive effects of the extracts also vary at different doses. And several studies have demonstrated that clinical efficacy of ashwagandha extract for management of muscles. And volunteers in taking ashwagandha extract showed significant increase in both muscle size and strength. Furthermore, a reduction in exercise-induced muscle damage was also observed. So this result suggested potential of ashwagandha as a sports supplement or medicine, but the molecular studies in muscle differentiation still need to be demonstrated. And mature skeletal muscles are composed of multi-nucleated muscle fibers called myotubes and operate considerable regeneration ability, which is performed by quiescent satellite cells. Once satellite cells are activated by appropriate stimuli, they undergo differentiation process, which can be characterized by expression of muscle-specific proteins, withdrawal of cells from the cell cycle, and their fusion into myotubes to repair injured skeletal muscle. A disorder of this repairability also occurs in various pathological conditions, such as cancer, Parkinson's disease, or even obesity and aging. And we all know that skeletal muscle is the largest tissue of the human body and plays an important role in maintaining homeostasis, making muscle differentiation an important target for treatment. So in our study, we investigated the effect of ashwagandha extract on muscle cell differentiation using C2C12 cell line, which is a well-established in vitro model. So we first generated extract from ashwagandha leaf and stem using an established method. And C2C12 cells were treated with either differentiation medium or differentiation medium supplemented with vitamin A or vitamin or ashwagandha extract for 7 days. And according to the morphology observation and the microscope showing here, we can see that among all the extract treated groups, they are from group 3 to group 12. Group 6, 10, and 12 showed stronger differentiated states, including elongated and widened cylinder shape and multinucleated myotubes. Then all the extracts were analyzed for the content of vitamin A and vitamin, which, as I mentioned just now, they are the main bioactive compound of ashwagandha extract. We analyzed the content of these two compounds by HPLC, and we found that extracts 6, 10, and 12 contain relatively low content of major vitamin lines and high ratio of vitamin to vitamin A as compared with other extracts. And we also subjected the control and treated cells to Western blot analysis for myogeny, which is a muscle-specific transcription factor involved in skeletal muscle differentiation development and is essential for the proper muscle differentiation. As shown here, groups 6, 10, and 12 cause stronger induction of myogeny expression, and which was in agreement with the strong differentiation observed under the microscope. Here, the imaging analysis by immunocytostaining was also in agreement with this result, suggesting that ashwagandha extract promoted C2C12 differentiation through upregulating the expression of myogeny. And protein aggregation is one of the causes of age-related decline in differentiation capacity and is closely related to neurodegenerative disease. We examined if ashwagandha extract could recover or reverse some of such damage by using two model assay systems. One is heavy metal-induced aggregation of GFP protein, and the other one is heat-induced folding of luciferase protein. So as shown in Figure 1, sodium arsenite caused the aggregation of GFP, and cells treated with methanol and the extracts that contained relatively high content of methanol showed maximum deaggregation. And of note, this result matched with the differentiation potential of extracts. And the quantitative measure of luciferase activity in cells subjected to heat shock shown in Figure C revealed heat-induced misfolding of luciferase protein. In contrast to the GFP aggregation assay, Lucifer's activity was increased more significantly in extracts 3, 7, and 11 that possessed a relatively high level of recyclin A treated cells. This data demonstrated that ashwagandha extracts protected cells against stress-induced aggregation of proteins. And differentiation of myoblasts requires functional degradative systems, including autophagy, to facilitate the elimination of pre-existing structures and proteins in order to assist the inflammation of multinucleated terminally differentiated myotubes. In view of this, we investigated the effect of ashwagandha extract on autophagy marker LC3B type 2. And Western blot analysis of control and treated cells revealed an increase in LC3B type 2 expression, especially in cells treated with extracts 10 and 12. And this result was also confirmed by immunocytostaining, suggesting that ashwagandha extract could promote cell differentiation by regulating
Recording in progress.
Sorry, Prof. Muhammad Yunus, you are muted and something wrong with the Zoom.
Okay, you hear my voice?
Oh, yes, we can.
Okay, you hear my voice?
Yes.
Okay, and after challenge.
Sorry, Prof. Yunus, something trouble with our breakout room.
Ah, really?
Yes. Sorry. Also, wait a minute. The committee will fix it.
But in my Zoom is no problems.
Your screen disappear in the Zoom. Sorry for this trouble.
Yes. And in my connected is okay.
Yes, Prof. Yunus, your connection is okay.
Okay.
Our Zoom is
I see.
trouble. Please wait.
Okay.
Sorry, we are so apologize.
Yes. Okay. It's okay.
Yes. Please join again to room 1.
Join again in room. And there is instruction over that.
Now you are in the main room, so
yeah. Sebentar. Yeah. Wait for a moment. The Zoom is.
Prof. Yunus.
Yes.
Can you see breakout rooms menu in this main room? You can find breakout room.
Iya Bu, ini kelas sendiri.
In the breakout rooms.
Iya
Prof.
ini.
Yunus.
Yes.
Yes.
But I don't know. I don't know to break. Okay.
Rista.
I think
the menu of breakout room is.
I live first and then I introduce
Prof. Yunus to join breakout room 1.
Okay, join first. Okay.
Join, yes.
Iya Bu, ini
join
saya.
room 1.
Ya, sudah Bu, ini Bu.
Iya.
Macam mana? Bukan ini loh, kayak gini ya. Aku custom juga tetap di sini.
Lagi sih nggak sistem tuh.
Sampai muncul pun harusnya dengan orang tersebut. Non-session semua tadi. Naik turun kan? Saya manajer.
Oh.
Nawasi lah. Curah itu ini nggak ada sekali. Kayaknya level bawah. Kembali ke main room. Ke main room lagi. Room dulu. Kamu ada sebelumnya? Lebih mau besok? Iya ya. Halo Mbak. Halo. Mbak Iki kok mau breakout roomnya itu close ya Mbak ya? Ngomong tiap menit. Tiap menit itu. Hah? Menit nanti keluar. Apa Mbak?
Kadang memang seperti itu.
Oh, terus berarti ini balik lagi ke main room gitu ah?
Kalau gini, kalau waktu ke close tiba-tiba ke close, itu langsung agak-agak izin apa jenenge di open lagi.
Open all room kan? Oh iya iya. Iya.
Sudah langsung masuk semua kok.
Oh ya sudah Bu, sudah saya open, sudah join semua ya.
Kadang memang seperti itu.
Oh ya. Sudah disuruh open all rooms lagi Bu. Lah sudah ini. Sudah itu lagi kembali lagi. Oh berarti open all room lagi ya Mbak?
Jadi misalkan ngono maneh langsung agak-agak di open all room.
Oh iya iya. Soalnya tadi itu kan open all room lagi, yang room 1 itu apa namanya? Banyak yang mau join gitu loh, Mbak. Jadi tetap gak bisa tak pindah.
Oh, ya berarti orangnya belum.
Enggak ada.
Budian, Bu Maratu, mohon segera join. Sudah saya masukkan ke room, tapi belum diklik join ya Bu, sepertinya ya.
Joinnya di mana ya?
Ada invite-nya Bu.
Enggak ada invite nih Mbak. Coba deh. Tadi keluar. Tadi keluar, sekarang nggak ada invite-nya.
Oh ya Mbak, enggak tunggu lagi Mbak. Apa sih Mbak? Ya.
Nggak muncul nih ya?
Nggak muncul itu invite-nya.
Nggak ada. Tadi sih ada, terus saya terus sekarang nggak ada.
Nah, saya pindah dulu ke room 1 ya, Bu.
Iya.
Ya. Sudah, ada.
Belum ada, kenapa ya?
Oh,
Ya, sebentar.
Oh, sudah.
Sebentar nih kayaknya ada deh.
Bu
Oh, nggak ada.
Bu Maratu saya pindah dulu ya ke room 2, nanti terus saya pindah lagi.
Oh, ada nih, udah.
Bu Maratu? Iya. Ini main room ya. Tadi Bu Emma udah tak jadiin call. Ini yang Hendra. Ya. Kamu koyo ini. Main room ya. Iya, Hendra. Coba Hendra. Langsung aja ya. Mohon segera join ya. Gimana Bu Maratu? Belum bisa join ya, Bu? Masih mute, Bu. Masih mute, Bu. Bu Maratu.
Tadi kan udah.
Iya.
Muter.
Iya.
Nggak masuk, Mbak.
Bentar, saya pindah lagi ya.
Tadi...
Ibu room 1 kan ya harusnya?
Iya, harusnya room 1. Terus tuh, nih invite you to join.
Iya, jadi masuk dulu ya. Berarti aku room 2 gak bisa masuk aku. Oh bisa sih ya. Nanti join sih ya. Iya, tapi kalau misalnya iki breakout utamanya dia tadi itu kayak gitu. Mohon maaf untuk yang untuk Bapak Muhammad dan Muhammad Fahmi dan Jelita.
Untuk Jelita dan Muhammad Fahmi bisa langsung join, klik join untuk di setiap roomnya.
Recording in progress.
And in this study, you use a male SDT fatty rats. And my question is, did you compare the pathophysiology with a female rat?
Yes. Several gender differences have been reported in SDT fatty rat, such as kidney, bones, or glucose and lipid abnormalities. And so, therefore, we hypothesize that there are gender differences in the brain as well. So, and we are currently investigating that. Please wait our result.
Okay, thank you very much. I wait for your next presentation next year. Okay, any questions from audience? Machiya Sensei, do you have any question? No? Okay, may I have a simple question to Dr. San?
Yes, please.
Your study is about muscle cells of extracts from ashwagandha. My simple question is ashwagandha used for treatment of muscle disorder in general?
Yes, there are some clinical experiments showing the positive effect of ashwagandha for treating, for managing muscle size and strength. And so we want to work on this more from the molecular mechanism side.
Okay, thank you very much. That's a very interesting presentation. Thank you.
Thank you, Sasase, as moderator. Next year you must be as a keynote speaker, isn't it?
Okay. No more questions from audience. Okay. Okay, let's move on to the next presenter. The next speaker is also from National Institute of Advanced Industrial Science and Technology, Japan, Dr. Marika Kurana. Our title presentation is Identification of Tectorigenin as a Hypoxia-Inducing Phytochemical, Potentials in Laboratory and Clinic. Dr. Kurana, please begin your presentation.
Okay, I will share my screen. Can you see my screen?
Yes, it's okay.
Just a second. Can you hear the voice? No. Okay, please give me a second.
We can hear your voice.
You can no the just a second.
Yeah, we hear your voice and also your presentation.
Can you hear my voice?
Yes.
Yes, okay.
Okay, you can continue, Malika.
Okay, so I will talk about the identification of pictorigenin as a hypoxia-inducing phytochemical and its potentials in laboratory and clinic. So what is hypoxia? Hypoxia is made up of two words, hypo and oxia. So basically when there is less supply of oxygen to the cells and tissue, it's called as hypoxia. Hypoxia in the recent times has highly been implicated in cancer and neurodegenerative diseases. So we hypothesized that hypoxia manipulation as treatments can be used as a treatment strategy by the use of phytochemicals. So how is hypoxia regulated on the cellular level? The mediator of hypoxia is hypoxia-inducible factor in the cells. When there is normal supply of oxygen, prolyl hydroxydehydratases hydroxylate this HIF1 alpha, and then it's ubiquitinated via von Hippel-Lindau factors and degraded. Whereas when there is less supply of oxygen or hypoxia, the HIF1 alpha is accumulated in the cytoplasm and then is transferred to the nucleus, where it binds with the hypoxia responsive element HRE and gives multiple effects via various messengers. So we carried out and we generated stable cell lines expressing HIF1 alpha driven luciferase reporter. We transfected 3 healthy cell lines with the transfection vector PGL44.2, and then we selected these HRE transfected cells by the use of hygromycin. After some time, the transfection check was carried out for stable cell lines, and the stable cells were cloned. And these HRE cell lines were further used
Recording in progress.
Terlempar lagi gimana ini?
In room 1.
Ini nunggu berarti ya?
Assalamualaikum warahmatullahi wabarakatuh. Ibu ini udah masuk di ruang kamu?
Main room, Mbak.
Belum. Ini masih di main room.
Saya bolak-balik masuk main room nih.
Ini gimana?
Breakthrough-nya di mana?
Nah, itu sudah ada room 1 join.
Recording in progress.
Hello, welcome back everybody. Should I start again?
Yes.
Okay, so after generating these cell lines, as I told you before, we seeded these cell lines into 96 well plates and we treated them with various chemicals from our AIST drug library, chemical library. We found out that CL6 or chemical library number 6 elicited a significant dose-dependent HRE response. After that, we wanted to see the toxicity levels, and therefore we again seeded U2S cells and found out that low doses of this drug was non-toxic. So now we use the EISD chemical drug library. Then we use the HRE-driven luciferase reporter assay and MTT cell cytotoxicity assays to finally understand that CL6 at low doses produced the HRE-driven luciferase response. Then what we did was that to confirm this HRE response, at the protein level, we carried out Western blotting. And again, as we can see in the Western blotting results, CL6 produced a dose-dependent response in HIF1 alpha. We also carried out immunocytostaining of these transfected cells with luciferase antibody to look at the dose-dependent response. Further, we wanted to check the cell viability in different cell lines. So we took U2S, osteosarcoma, TIG, human lung fibroblasts, HD1080 fibroblastoma cell lines, and MCF7 human breast cancer cell lines to carry out short-term and long-term viability assays to understand that CL6 was non-toxic. So here we propose one potential application. We know that conventionally, cobalt chloride is used as an in vitro chemical model to induce hypoxia. But it suffers from toxicity at hypoxia-inducing doses. Also, it is a metal compound. And the mechanisms of actions are not completely understood. We also now know that incubator-induced hypoxia and cobalt chloride-induced hypoxia activate different signaling pathways. Therefore, we can postulate that CL6 could serve as a non-toxic and safer alternative to cobalt chloride for studying hypoxia signaling, since it produces dose-dependent HIF1 response as compared to cobalt chloride. Well, to confirm this phenotype of hypoxia induction, because we know that hypoxia also induces cell migration, we used U2S cells and carried out a cell migration assay. As we can see here, with the increase of time and dosage of CL6, the migration of cells increased. We also wanted, because hypoxia-mediated cell migration also is activated and mediated by MMP9, we did a Western blotting experiment and found out that MMP9 was upregulated in CL6 treated cells. With literature survey, we know that MMP9 is involved in the degradation of extracellular matrices, which is further involved in embryonic development, reproduction, angiogenesis, bone development, wound healing, cell migration, learning, and memory. Some researchers also show that the role of MMP9 in neuronal plasticity and neuroprotection is marked. Neuronal MMP9 has participated in synaptic plasticity, thus playing a pivotal role in learning, memory, and cortical plasticity. We also know that moderate amounts of MMP9 elevation is known to have neuroprotective role in Alzheimer's disease mice. Thus, we investigated differentiation potential of CL6 using brain-derived cells. So we took C6 glioblastoma cells and we established a differentiation model by treating these cells at low doses of CL6 for 20 days. Here we can see in the phase that the differentiation is marked. We can also see that markers like GFAP and PSE95 are upregulated. So what is CL6? CL6 is also known as dictorigenin. It is an orthomethylated isoflavone, as we can see the methyl group attached with the O group in the flavonoid moiety. It is derived from the rhizomes of leopard lily, which is formerly known as Iris domestica from the family Iridaceae. It is flowering plant perennially growing on the hillsides in East Asia. Some known activities are anti-inflammatory, inhibition of NF-kappa B-AKT cytokines, COX-2 and INOS pathways, antioxidant by attenuation of mitochondrial membrane potential, and ROS quenching. It also has a regulation of PPAR gamma and has shown release of erythropoietin and HIF1 alpha induction in neurons. So what is the take-home message or summary? CS6 is a candidate phytochemical that can be recruited for manipulation of hypoxia and hypoxia-related stress and neurodegenerative disorders. At the end, I would like to thank AIST India Lab, MEXT, the Ministry of Education, Culture, Sports, Science and Technology, Japan, for sponsoring my study, and the University of Tsukuba. Thank you, everybody.
Okay, thank you, Dr. Kurana, for your great presentation.
I can't hear you, I'm sorry.
Okay, next presenter is from University of Brawijaya, Indonesia, Dr. Titin Andri Wihastuti. The title presentation is Platelet Cells, Mediators of the Atherogenesis Inflammatory Response in Atherosclerosis Risk Groups. Dr. Wihastuti, you may begin now.
Till 6 hours, 48 hours, and 60 hours. And then the tempe edamame manufacturing process. First, fresh edamame is done stripping the skin, then washing the seed of edamame or mukimame, and then after that soaking for 9 hours, then boiling with a temperature of 100 degrees Celsius for 5 minutes, and then stripping the skin of edamame seed, then drying and add inoculum according to the treatment. Then in the container on plastic that has been give a hole, then store at the room temperature 28 until 30 degrees Celsius. And the step 1, after getting the after the process of making according to the treatment of inoculum concentration of 0.5%, 1%, and 1.5%, and the lack of fermentation for 36 hours, 48 hours, and 60 hours. After becoming a product of tempe edamame, chemical analysis is carried out, which include water content as fat, fiber, and protein, and isoflavone. In the step 2, after getting the best treatment from step 1, tempe edamame treatment is the best with commercial derived from Sanan and from Singosari area, which is the test to find out the amino acid and bioactive peptide of each product. And the result is chemical analysis. Based on the result of Celeni calculation, the combination of the 48-hour fermentation treatment and the inoculum concentration of 0.5% due to the long fermentation of the 48 hours, and the inoculum concentration of 0.5% was chosen as the best treatment because the nutrition content obtained for the following table data obtained the highest protein and isoflavone content. The content of water, as fat, and fiber is still lower compared to the other treatment. And then amino acid. Furthermore, in the following table, is the result of analysis in the second stage, where tempe edamame treatment is the best compared with tempe from Sanan and Singosari. To find out the amino acid content is at sample which is as follow. The result of amino acid identification from all 3 samples in the study also show the content of amino acid L-glutamic acid is higher among other amino acid. This can make tempe become more savory. The higher the content of glutamic acid, the more savory the product. And after that, there is bioactive peptide content. Based on the following figure and the table are the result of SDS page to find out the bioactive peptide present in tempe. From 3 samples of tempe, it is make the tempe edamame and Sanan tempe are not perfectly hydrolyzed, result in the 8 band on tempe edamame and 6 band on tempe Sanan. While tempe Singosari is perfectly hydrolyzed, resulting in 10 band. From each of this band is a bioactive peptide obtained from the tempe product. And the conclusion based on the result that has been carried out, conclusion can be drawn as follow. The best tempe edamame treatment is found in the old 48 hours fermentation treatment and the inoculum concentration of 0.5%. Tempe edamame has amino acid content and bioactive peptide content lower than Sanan soybean, tempe, and Singosari sementing. Maybe thanks for all, and back to moderator.
Thank you very much, Ms. Fitria Faradila. We will begin the discussion session. There are several questions in the chat room. First question, 2 questions for Professor Muhammad Yunus. The first question is how this Emiria maxima oocytes efficiency compare commercial drug
Recording in
treatment,
progress.
and how this Emiria maxima oocyte protein efficiency compare commercial drug treatment in coccidiosis treatment. It can be as prevention treatment. For Professor Yunus, would you answer this question? You're still unmute, bro, your microphone.
Okay, I would like to clarify. I still inside in the room.
Okay, yes.
Okay.
Okay,
Okay, good
1.
question for Dr. C. Arko Pradhanto. Yeah, this is from this question. I would like to share the little information about if we compare about the efficiency if we use the Emiria maxima. As we know, the Emiria in the chicken coccidiosis, there's several species. You know, one of the species of Emiria maxima, and the characteristic of Emiria or coccidiosis in the chicken is species specific. It's mean if we use some species, if we use the species Emiria in coccidiosis vaccine, it's better we use the comprehension species, because if the chicken infected by one species not resist in the other species, it's better comprehension species around 7 until 9. But this is consideration based on the location and the area of the farm. Because some location in different species, but little bit difference is of the number of Emiria species. It's mean, you know, why we explore about the vaccine, because the vaccine are one of the good prevention in the farm, not only in the chicken, but also in the ruminant farming, like dairy cattle or beef cattle. If if 2013, the WHO have the restriction if we use the chemical agent to use the animal production, and this is we have to consider this one, you know, because the vaccine is very good to prevention some diseases. And then if we compare about the Emilia maxima, if we compare about the commercial treatments, I think not equal, because even now in our farming by the feed mill, like pork palm, like confit, like some and so on, Cargill, they still use the little bit chemical agent. But the WHO have big restriction, but to avoid and prohibited if we use the drug in the animal production. And this is the good opportunity in the good consideration if we use vaccine. But if we use vaccine in the coccidiosis in poultry, we use the comprehension species around 7 until 9 species. Is the good solution to prevent coccidiosis. Just one of species Emilia, and the other species one species of Emilia maxima, and the other species is Emilia tenella, Acerculina, and also Necatrix, and so on. But if we compilation one another and we use the kill vaccine, is better if we compare, because some the prohibition our export to the other country, it means we still use the chemical agent. This is very bad effect on our export material, not only the animal production and also the other production. And the other question, if the efficiency, one more again, if we compare about the material vaccine in coccidiosis poultry, be compilation about some species, not only just one species, because Emilia species in chicken, there is 7 until 9 species, and the characteristic is the species specific. Just one species resist one species, not the other species, and then the comparison one another in one material, I think is better. I think that's all information for me. Even our farming still a little bit used, because this is our problem in our society in the farming, like the tradition, particularly in the traditional farming, like the broiler or poultry or layer. I think if we compare about drugs, it's better we use the vaccine, even kill vaccine or attenuated live vaccine. But attenuated or live vaccine is the high risk to the progenic or pathogenic form again, because the environment to be affect the growing up about the pathogenic, pathogenic again. Thank you.
Thank you for the answer, Professor Yunus. We move the next question for Fitria. What is the special characteristic and lack of product tempe edamame from preference consume, and the advantage of edamame tempe compared and beside the nutritional content when compared to soybean tempe? Can this edamame tempe consumed by someone with certain diseases? And the difference between edamame tempe and commercial tempe used to affect the nutritional content. Okay.
With the ingredients, tempe consumes someone with certain diseases. With these ingredients, consuming edamame can help lower blood sugar level and is also consumed by people with a high blood pressure. Edamame also contain an antioxidant that are good for maintaining the immune system. And between edamame tempe and commercial by effect and nutrition content, the difference between tempe edamame and commercial tempe is the type of inoculum used. Inoculum used in tempe edamame is a Raprima type inoculum, Rhizopus oligosporus, and commercial tempe using conventional or traditional type of user. This user has many type of unit. Thank you very much.
Yeah, thank you very much, Mrs. Fitria Faradila. The next question is for Professor Yunus again.
Yes.
Okay. In the column of chat, is it worth for the farmer in Indonesia use these vaccines because the cost of poultry is big in reality?
Yes.
Professor.
Okay, thank you for Dr. Asri Aku. Yeah, if our farming in our society in the farming like layer or broiler, I mean about in broiler is no problem because the broiler lifespan of broiler is not so long, just 32 days. After 32 days, we have harvested about the broiler. But in the problem is the layer, you know, in the layer, just the like the modern farming like closed house or something in like in the farmer, but to follow about the modern farmings, I think they use the vaccine or coccidiosis. But in the traditional farming, the traditional layer farmings, I mean about they still follow about the maybe sometime to follow about the feed mill products. I think like feed mill, like the pork pan, like confit, like carcass, and so on. And yeah, because now is the price of coccidiosis vaccines is still so expensive. I think this is the problem our farmer. But if we have short consideration and short opinions, I think look like expensive. But if we think about long term in the future, I think this is the step by step and gradually the good effect, the good impact on our farming. Because if we use the drugs to prevent about the coccidiosis, I think so many complicated bad impact, you know, like the resistant residual products in the meat or in the eggs. This is its problem. But some several part in the farming.
I'm sorry, Prof. 1 minute left for answer.
Okay. Okay, thank you.
Is it all the answer or?
Yes, I think yes.
Okay,
that's because business as the real thing in the field, in our farmer in layer or broiler. Yes.
Okay. That's all the complete answer, yes, bro. Okay,
yes.
Thank you, bro. Okay.
Yes.
Due to the limitation of time, we will move to last session for presentation. We will for Ibu Maratu Soleha will present two paper about identification and anti-cancer testing result of Salpinia sapan subcritic extraction and anti-cancer activity and active compound of Siniber serum bed. Okay. Please welcome Ibu Dr. Maratu Soleha.
Just a moment, I'll try to.
All right, good afternoon to all participants and the committee. First of all, I would like to thank you for having me in and give me time to present my research. First of my research, I would like to present the title Identification and Anti-Cancer Activity Test of Salpinia sapan Subcritic Extraction. The background from WHO data, the cancer prevalence in 2013 was 0.4%, and the data increase into the incidence increase in 2018 become 1.8 per mil. So it increased around 0.4 per mil. Yeah. The largest cancer is breast cancer, and followed by the prostate and lung cancer at the second and third. In Indonesia, based on national survey, the incidence of or the prevalence of cancer from the doctor's diagnosis in 2018 was 4.62 per mil. In the age group 50 until 64 age group. Yeah. The cases were higher in women compared to men. So as we know, the FDA has been approved the drug for healing cancer. Some of them come from herbs and a plant. Yeah, for example, vincristine and vinblastine has been used for lymphoma Hodgkin cancer. Both of them come from Vinca rosea herbs. And also the other one, well-known drug, paclitaxel and docetaxel, it's also come from plant Taxus species. So the other one is campoterine is also metabolite that has been extracted from Zingiber santorisa, ya, from Curcuma santorisa. Curcuma santorisa in Indonesia can grow, so it can be found in many places. So for this experiment, I choose herbs. I choose plant Caesalpinia sapan. Caesalpinia sapan, woods and seeds has been proven to effective against some cancer. It has been tested against MCF7 cell line, and it has value IC50 29.98. And for the seeds, known has been for the seeds, ethanol fraction is also effective against cancer. Have IC value quite low, 28.8. For the method of the experiment, I will deliver into 3 parts. First, extraction. The second, cell toxicity. And the third is prediction active compound. The extraction method is different from the common experiment. This extraction method, I use subcritic extraction. Normally we extract the pulverized plant or herbs with solutions such as ethanol or methanol, or the other one hexane, etc. But this one we didn't use the solution. we just flow the gas through the pulverized herbs, and the gas will change from the solution form to the gaseous form by the change of the temperature and pressure. Yeah. But before we extract, first we did the drying of the woods of Caesalpinia sapan by the hydrator to reduce water. To reduce the we drying first at the temperature 60 degrees Celsius for 24 hours. After that, we running in the extraction tool subcritical using HFC R134 extraction gas with the extraction temperature around 31 to 27 degrees Celsius, and the pressure 10 to 9 bar up and down. The second part of my experiment, I did a toxicity test. I did this test with MTT assay. MTT and the cytotoxic activity of the sample we observe by observing the viability of the cell. A little bit method of MTT assay pathway is MTT will react with the enzyme succinate dehydrogenase in mitochondria of the living cell, and MTT and this enzyme will react and form the formazan crystal, and we observe this formazan crystal in the wavelength 570 nanometer with ELISA reader. This one serial concentration of the extracts against MCF7 cell line. So we observe this absorbance of the wavelength, and we make the standard curve or regression curve to measure the IC50 value of the sample. And the last one, the last method is predicted active compound. Testing using chromatography tools, GCMS, with the specification as shown in the slide. And here come the result and discussion. The yield of subcritic Caesalpinia sapan extract was 0.03%. This is quite low compared to the extraction method with the solution using 35% of ethanol. The yield was 25%. And the AC value from this experiment, AC value sapan against MCF7 cell line is 38.288 gram per milliliter. And also we from the GCMS shown it has 15 kind of metabolite or substance. So this one, the regression curve the extract again MCF7 to measure the AC50 value of the sample, and in vitro on other cells such as human leukocyte, yeah, sapan caesalpinia sapan is effective in other cells such as HL60 and also HeLa, human cervical adenocarcinoma, Lewis long carcinoma cell, and has been carried out also. It used caesalpinia sapan seed, compound that play a role in inhibiting growth of the cells are paginin D and also many kind of paginin and also brazilin. Yeah, but for it can be proven from the other research that caesalpinia sapan is effective against cancer cell line. So that is end of my presentation.
Okay, it is the first presentation from Ibu Maratu. We will continue for the second presentation by Ibu Maratu for the second paper. Ibu Emma, would you play the second video?
Sorry, Dr. Nia, she wants to present live by herself.
Oh, by herself.
The group WhatsApp.
Oh, okay. Is Ibu Maratu is already in the room 1? No.
In the WhatsApp group, she said that
Ibu Maratu,
she couldn't join in the Zoom.
but her name is here, Maratu Soleha in room 1.
Ibu Maratu.
Ibu Maratu. Okay, meanwhile, we wait for the Ibu Maratu second presentation.
Ibu Maratu, please unmute your microphone in the Zoom.
Okay. Hello. Ada enggak sih ibunya?
Ibu Maratu say in WhatsApp, she only can hear the voice, but she cannot speaking.
Oh, your microphone is remain mute, Ibu Maratu.
She didn't see the screen. Ibu Maratu, please, you check your Zoom at the button, yeah. Microphone button ya, Bun ya?
Yeah, there are microphone icon in the left bottom. You can click and then you can speak. Oh, atau izin Yura presentasi dulu.
Okay, Yura. I will play.
Ibu Maratu, we apologize for this inconvenience, but we will continue to the last presenter. Meanwhile, we wait for your connection is stable. Okay, Ibu Maratu. Okay, please Yora for Yora Dwi Wibangga. You ready for your presentation?
I will share the pre-recorded video from Yoga.
Okay.
Yeah.
Okay. The committee will play the video. Thank you, Ibu Emma.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Hi, hello. My name is Yorafika Dwi Wibangga from Brawijaya University. I would like to present our research. The title is Identification, Characterizations, and In Vitro Antioxidant Activity from East Java Red Rice Bran. And this is the research we've been working on, so let's get started. Rice is the most popular member of the Poaceae family. Worldwide, more than 40,000 varieties of rice are cultivated. The amount of rice productions and consumptions in the world has increased almost 6 times. In 1960, with a total of 7.5 million tons, increased to 44.2 million tons in 2015-2016, mainly in Asia. In Indonesia, the amount of rice productions and consumptions has also increased. In 2020, productions increased by 1% compared to the previous year, which reached 31.63 million tons. In general, white rice dominates the rice market, but lately, people are also interested to consuming pigment rice, one of which is red rice. Red rice contains secondary metabolites that have positive health benefits, such as phenolic, flavonoid, proanthocyanidins, anthocyanins compounds, which have biological functions as antioxidant, anti-diabetics, and anti-cholesterol. Rice bran has the potential to be explored for their contents and biological activities. Red rice bran contains flavonoid, phenolic, anthocyanins, proanthocyanidins, tannins, alkaloid, and some essential oils. Bran is a waste of rice production that has not been utilized optimally as a functional food ingredient. Until now, its use is still limited as animal food, organic fertilizer, and alternative fuels. To the best of our knowledge, explorations of biological activities from red rice bran from East Java has not been conducted. Red rice bran, there are several flavonoid compounds, namely quercetin, apigenin, catechin, luteolin, and myrcene. Flavonoid and phenolic compound are a group of secondary metabolites that have an aromatic ring and at least one hydroxyl group. These flavonoid and phenolic compounds have potential as antioxidant because they can donate one electron to free radicals to stabilize free radicals, so that can protect cells from damage caused by free radicals. So, based on the introductions, the objective of this research to identify, characterize, and to investigate antioxidant activity of the extracts from red rice bran from East Java through phytochemical analysis, FTIR, and antioxidant test. The materials used in this research are red rice bran varieties of Mentikwangi, Aik Sibundong, and Blambangan from the method used are macerations of bran powder with methanol solvent, qualitative phytochemical analysis to identify certain secondary metabolites, determinations of total phenolic and flavonoid content, antioxidant activity test by FRAP method, FTIR analysis to determine the functional group present in the bran extract, and statistical analysis with ANOVA one way. from phytochemical analysis revealed that the red rice bran of Mentikwangi, Aik Sibundong, and Blambangan contain of flavonoids, tryptopanoid, phenolic, tannin, and glycoside, whereas the taste for steroid, alkaloid, and saponin of Mentikwangi and Blambangan, and then alkaloid and saponin of Aik Sibundong yielded negative results. From the measurement results of the absorbance value, the higher absorbance indicates the higher concentrations of secondary metabolites contained in the brain extract. Tryptopanoid and flavonoid were considered the main place of secondary metabolites identified in compound known to have high antioxidant activity. From total flavonoid and phenolic content taste, the highest total phenolic and flavonoid content are in Mentikwangi red rice bran. The potential of phenolic and flavonoid compounds as antioxidant are caused by the hydroxyl groups in phenolic and flavonoid compounds. The hydroxyl group functions as a hydrogen atom donor when it reacts with radical compounds through an electron transfer mechanism so that the oxidation process can be inhibited. And then antioxidant activity analysis with FRAB method. The FRAB method is often used to evaluate of an antioxidant to the presence of antioxidant in the extracts resulted into reductions of the ferric cyanide complex to the ferrous cyanide complex. The reducing power was found to be in all acid stronger than Sibundong and stronger than Mentikwangi. The antioxidant activities of all red rice bran ascorbic acid with IC50.28 microgram milliliter. A lower IC50 value represents a stronger free radicals inhibitor. Based on the IC50 value, Blambangan bran and ascorbic acid were classified as very strong antioxidant, while Mentikwangi and Aik Sibundong bran were classified as weak antioxidant. The FTIR spectra of the samples aren't visually different, showing that the chemical components content are generally same. FTIR result confirm the result of phytochemical analysis. Based on figure 2 and table 4, major compounds that have been found contained in the 3 red rice bran are some specific functional group, which are hydroxyl, alkane, aromatic, ester, alkene, alcohol, carbonyl carbonate, ether, and ketone. So, the conclusions of this research show that each red rice bran has specific phytochemical content and functions as an antioxidant. FTIR results confirm the results of phytochemical analysis. The highest antioxidant activity was found in Blambangan red rice bran with an IC50 value of 1.09 microgram milliliter. This research was partially funded by the LPDP from the Ministry of Research and Technology and Ministry of Finance, Republic of Indonesia. So everybody, do your best at every opportunity that you have. If plan A didn't work, the alphabet still has 25 more letters. I want to thank you everyone for your undivided attentions and for listening to my research project. Thank you. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you for your Rafika for the nice presentation. Now we will back to the Dr. Maratu Soleha for the second presentation. Hello, Ibu Maratu.
Yeah, okay. I'll try to share screen.
Okay, you will share screen by yourself, okay? Right?
I try to open. I have opened it, but where is it? Oh, sorry. I tried to open. Sorry, I detect it's the PPT is already on my. I cannot in the share screen. In the share screen here, can you please help to share?
Okay.
May I help you to share your PPT or your pre-recorded video, Bu Maratu?
Just PPT, okay.
Just PPT.
PPT will be okay.
Okay, the committee will help you to share the PPT to screen and you will present by yourself directly.
Yeah, I enrich the PPT for the update, but I'm sorry I cannot show to you all.
Okay, no worries. Bu Emma has started share for your PPT. Okay, you can see the PPT clearly. Ibu Maratu.
Yeah, yeah.
You can start your presentation now for 10 minutes. Thank you.
Thank you for the second chance of sharing my research. This is my second talk. It's still same. My research still explore about anti-cancer activity, but the second talk is come from the other plant, which is Zingiber zerumbet. Okay, next please. Next, please.
Sorry, something trouble. Wait.
Yeah, I didn't go through again to the background because, as we know, from the keynote speaker, that the incidence of cancer, of course, still high, and the drug is still is not safe. Still, we need to explore the alternative drug to heal the disease. So the second plan that I try to explore is Zingiber.
Recording in
No
progress.
studies have yet described the correlation of these two molecules. So, this study was conducted to investigate the relationship of LPPL-E2 and VEGF levels to the severity of patients with risk of atherosclerosis, which assessed based on the Framingham scoring risk system, or FRS. In the next section, we will discuss about the research methodology. The study was a cross-sectional study consisting of 158 respondents. The study was designed in a non-probability sampling method using human venous blood samples taken from population at risk of atherosclerosis grouped by the FRS. Measurement level of circulating LPPL-E2 and VEGF carried out at the Central Laboratory of Saiful Anwar Hospital, Malang. Analysis of blood samples was done using ELISA according to manufacturer's protocol. Okay, the next thing I will share with you is the result of the study. The baseline characteristic for the studies consists of several characteristics. First, about the gender, the majority of the respondents are male with 60.8%, and the other are female with 39.2%. Most of the respondents are 60 to 64 years old, occupying 24.7% of total respondents. And most of the respondents also do not have a smoking habit. And the majority of the respondents have normal blood pressure, normal lipid profile, but most of the respondents are categorized as obese according to their body mass index. There is stratification based on FRS in patient observed in 3 levels: low, intermediate, and high risk group. We can see the result in table. Also, data analysis using person correlation test showed that the concentration of LPPL2 had a strong correlation with VEGF expression. It also showed a positive correlation, means LPPL2 concentration will increase along with VEGF concentration, vice versa. However, concentration of VEGF and LPPL2 did not match the FRS group, means it does not follow the stratification of the FRS. Okay, next we are going to discuss about the study result of LPPL2 and VEGF. So, LPPL2 is a secreted molecule by inflammatory and non-inflammatory cells such as lymphocytes, macrophages, mast cells in plasma. LPPL2 used as a biomarker will significantly increase in which it represents the incidence of vascular inflammation. It also associated with the pathogenesis of plaque development in atherosclerosis. Therefore, increased LPPL2 are independently associated with heart disease severity and could differentiate patients at high risk of cardiovascular events. This study explained that the concentration of LPPL2 obtained in the low risk group is 0.1017 micrograms per milliliter. It is the highest compared to the other groups. The discrepancy of the result against the clustering group of FRS is supported by clinical reports that low level of LPPL2 along with other conventional markers indicate a low risk of acute coronary syndrome. While other studies say that LPPL2 is not suitable for parameter in low-risk population. Several other studies also showed that LPPL2 assessment is only recommended for high-risk patients. It allows for further risk stratification among high-risk patients that require more aggressive therapy. Based on the American guidelines for assessing the risk of cardiovascular disease, LPPL2 recommended as an additional marker for classic risk assessment in moderate and high-risk patients. On the other hand, VEGF is a mutagen that promotes angiogenesis and vascular endothelial cell proliferation. It triggers angiogenesis, which plays a role in regulating integrity and is related to endothelial functions. However, it also increases the risk of unstable plaque and plaque formation events in atherosclerosis. In this study, VEGF acts as an endothelial growth factor in atherosclerosis incidence. It did not show a significant increase in patients according to the FRS. VEGF presence is physiologically a form of compensation for vasa-vasorum angiogenesis when vascular inflammation occurs. On this research, the average level of VEGF also showed the same value. We can consider that there is a physiological limit for compensation, and it is not concerning pathological angiogenesis. Correlation analysis of LPPL2 and VEGF level related to the increased function of LPPL2 when there is a vascular inflammation happens. Then VEGF will emerge to perform endothelial repair. VEGF can trigger formation of new blood vessels that are fragile and worsen atherosclerosis. Based on this result, VEGF and LPPL2 has a positive and significant correlation. Okay, that brings us to the end of the presentation. In conclusion, LPPL2 has a significant correlation with VEGF, so that it has the potential to be a biomarker of vasa-vasorum angiogenesis in atherosclerosis risk population. And I sincerely appreciate your attention today. If anyone has any question, please feel free to ask and I will do my best to answer. Thank you. Wassalamualaikum warahmatullahi wabarakatuh. Good afternoon.
Thank you for presentation from Bapak Boyono. The next one we will have Ibu Ika Octaviani. Ibu Ika Octaviani is here.
Yes, I am here.
And you already sent your video presentation. So we will play your video presentation. The presentation title is The Effects of Low Swim Training in Reducing Prostaglandin Plasma and the Expression in Sacroiliac Cartilage of Pregnant rat models. Ibu Ika is a doctoral program of medical science, Faculty of Medicine, Universitas Brawijaya, Indonesia. Okay.
Thank you. Assalamualaikum warahmatullahi wabarakatuh. I'm Ika Octaviani. I will presentation our study. Our study is the effect of low swim training in reducing prostaglandin plasma and the expression in sacroiliac cartilage of pregnant rat models. Increased uterus weight during pregnancy, move down of gravity forward, can increasing lumbar lordosis and excessive pressure on the pelvic gather area will frequently generate pain. Pain is commonly felt in the area of posterior iliac crest and gluteal folds, especially around the sacroiliac joint, and can also occur in the pubic symphysis area. Those complaints were felt doing activities such as standing up, walking faster, climbing stairs, turning in bed, changing sleep position, and even being severe disability. These pains commonly start to appear at the second trimester of pregnancy. The etiology may be associated with the increased weight of the uterus and increased relaxin hormone during pregnancy. This condition causes movement of the surface of SID so that it changes the dominant pressure position to one side of the SID. Pressure on the SID due to the burden of pregnancy is considered to initiate local joint injury, which further contribute to the release of various chemicals such as adenosine triphosphate and inflammatory mediators, one of which is prostaglandin PGE2. Prostaglandin sensitizes aberrant nerve terminal endings that are responsible for an increased response to pain stimuli and are a direct stimulation of primary afferents. This is our theoretical framework, and we can see the swim training have advantage are buoyancy and water viscosity. The other advantage is water hydrostatic pressure, and swim training can mechanical loading cartilage. The wall of the theory can be decreased prostaglandin 2. Swim training during pregnancy can reduce the burden that the body has to accommodate, which is expected to reduce the pulling pressure on the muscle and joint in the pelvic area. The existence of buoyancy, hydrostatic pressure, and the viscosity of water makes the muscle don't experience fatigue. If accompanied by training, water sport will provide a good mechanical load to the cartilage, which will activate the chondrocytic beta signaling, which of course will improve the cartilage structure. The study aims to bring scientific evidence whether the swimming exercise can down turning plasma prostaglandin and prostaglandin 2 expression in SIG pregnancy. As an experiment, this study was conducted in vivo Ratus norvegicus. Animal experiments, we use 36 female rats and 18 male rats, and we get ethical clearance from Animal Research Ethics Committee of Brawijaya University. Experimental study use a post-test only with a control group designs. And our experimental group are 6 groups. Study procedure, the first stage is the rat impregnation. We use synchronization of estrus base. And then the next steps, we give induction of relaxin and normal saline in pregnant rats. The dose of relaxin can capable to increase rerum relaxin level and within the diameter and provide cartilage structure modification in pelvic area after 24 hours of injection dose. The swimming training period starts with an adaptation process for 2 days of 10 and 15 minutes period. The adaptation process on the 9 and 10 days of pregnancy. Later, it's followed by 2 stages of core swimming training. Each stage had 4 days duration, which was separated by 24 hours as a resting day. Each of them was performed 60 minutes long. The blood was collected from the lateral A vein and surgical preparation. Before surgery, the rat was anesthetized using ketamine and using cervical dislocation. The process histological cartilage, we start by decalcification, which use an immune rapid cal fluid for 3 days. And immune histochemical reagent, we using prostaglandin 2 monoclonal antibody from Santa Cruz Biotechnology. Data were analyzed based on one-way ANOVA and Kruskal Wallis. This is our result. We can see the table 1, level of prostaglandin plasma on before the swim exercise. So the significant difference based on one-way ANOVA, and on after swim exercise, also significant difference based on Kruskal Wallis. The following are 3 pictures. We can look, the first picture is negative control. Negative control, so negative expression PGE2 in SIG. But the second picture is second positive control. We can see stronger and deeper expression in hyaline cartilage on left side SIG, as well as stronger in ligament on the left side, and also stronger in fibrous tissue, but negative expressions in right side ligament and right side hyaline cartilage. And this is the third picture. The picture 1 of swim treatment sample. The picture shows negative expression on the 2 side ligament, but still middle expression on the hyaline cartilage margin in the left side, and the weakness expression in the right side hyaline. Middle expression on the fibrous adjacent to the muscle tissue. This is figure of mean value expression of PGE2 in SIG based on the one-way ANOVA statistic. There was a significant difference in the expression PGE2 in all experimental groups. The study discovered that swimming exercise are highly potential used as complementary treatment as inhibitory therapies for increasing prostaglandin in pregnant rat. According to Almeida, regular exercise can relieve musculoskeletal pain and spinal pain as well. The Azambuja, his study proved short-term swimming exercise effectually prevents spinal cord sensitization. Floating in the water during light swimming session will give the relaxed sensation as the body lost its original weight, especially in the submerged body part. Another advantage of swimming training is the ability of water viscosity and hydrostatic water, which allows a change in the compressive force. The hydrostatic pressure of water can positively affect the cardiovascular system. Helma conducting a similar experiment that divides subject into training and non-training, which the training group a significant increase of IL-10. An increase in the anti-inflammatory cytokine IL-10 produced by chondrocyte will inhibit COX-2 and prostaglandin 2. This advances our research. This study was only applied to rat primigravida. The comparison between primigravida and multigravida pregnant rat needed to be observed. Our study provides evidence that pregnancy can increase prostaglandin plasma level and expression in the fibrous and cartilage tissue of the sacroiliac joint. We discover a significant difference in plasma prostaglandin level and expression in SIG in the group that performed swim train compared control group. Based on the research result, it is hoped that it will contribute to the further research on swimming training for pregnant women. Thank you for the attention.
Thank you very much for Ibu Ika Octaviani for your presentation. So now we will have the last speaker for this oral session. So we will have Ibu Sinta Oktiawardani from Faculty of Medicine Universitas Brawijaya Indonesia. The title of her presentation, Ibu Sinta is here, I can see you. Okay, the title of the presentation is Pathway Analysis on the Effect of FOXO3 ARS 4946936 gene polymorphism on major molecular responses of immunitative mesylate therapy for CML patient in Japanese based Indonesia.
Assalamualaikum warahmatullahi wabarakatuh. Can share screen. First, thank you for the Third International Seminar on Smart Molecule of Natural Resources, ISSMART, and Association Federation of Biotechnology, AFOB committee. I will present my study, Pathway Analysis on the Effect of FOXO3 RS 4946936 gene polymorphism on major molecular response of imatinib mesylate therapy for CML patient in Japanese race Indonesia. For the introduction, many studies have shown that advanced stage high blood cell count and high risk prognostic scores are associated with non-achieving a major molecular response of chronic myeloid leukemia patient receiving imatinib mesylate treatment. Genetic factors are thought to play a role in primary resistance to imatinib mesylate, one of which genetic polymorphism. The FOXO3A gene is a tumor suppressor gene in cancer. And the loss of function of the FOXO3A gene is associated with resistance to cancer treatment. Objective of this study aim is to determine which pathway is most influenced by the genetic factors FOXO3A RS46936 gene polymorphism on the imatinib mesylate treatment response. Methods, study design, and patient. This is a prospective cohort study at Saiful Anwar General Hospital between February 2019 to February 2021, and with consecutive sampling method of patient recruitment, and the study had been approved by local ethical committee and has been conducted following the Code of Ethics of Helsinki Declaration. This is the subject of this patient. The population is positive BCR ABL CML patient who came to Dr. Saiful Anwar General Hospital Malang with a sample CML patient with positive BCR ABL result who have received TKA treatment for at least 6 months at Dr. Saiful Anwar Hospital Malang. With the inclusion criteria, all the CML patient with positive BCR ABL treated imatinib mesylate at least 6 months in our hospital during the study period. The age more than 18 years old and providing the written informed consent to participate in this study. With the Japanese race with 3 levels above, parents, grandparents, and great-grandmother. With the exclusion criteria, patients were excluded from our study if they had the history of COPD, vitiligo, prostate cancer, breast cancer, and gynecological cancer. This is the baseline characteristic of patient included in our study, and there is 2 parameters, leukocyte and blast cell count, that statistically significant between all the genotype, CC, TC, and TT genotype. And this is a pathway analysis equation model with direct path coefficient and p-value, which is, I give the red highlight for the correlation that statistically significant, and the black highlight for the non-statistically significant. And this is the PET coefficient indirect effect on exogenous variable on endogenous variable or through antecedents variables. And there is no data that statistically significant. And this is for the total influence of PET coefficient in our study. The result is there is a statistically significant correlation between TT genotype, FOXO3A gene polymorphism RS494696, which is it increased the possibility of not achieving major molecular response. The PET analysis results show that directly TT genotype of the FOXO3A gene polymorphism RS494696 increasing the risk of not achieving a molecular response and statistically significant. And indirectly, a TT genotype of the FOXO3A gene polymorphism increase the risk of not achieving a molecular response, but not statistically significant through an antecedent variable leukocyte count. Indirectly, TT genotype of the FOXO3A gene polymorphism RS494696 increase the risk of not achieving a molecular response, but not statistically significant through the antecedent variable leukocyte count and clinical phase. Indirectly, TT genotype of the FOXO3A gene polymorphism increase the risk of not achieving a molecular response, but not statistically significant through the antecedent variable clinical phase. Indirectly, TT genotype of the FOXO3A gene polymorphism increase the risk of not achieving a molecular response, but not statistically significant through the antecedent variable blast cell count. And indirectly, TT genotype of the FOXO3 gene polymorphism increase the risk of not achieving a molecular response, but not statistically significant through the antecedent variable blast cell count and clinical phase. Indirectly, TT genotype of the FOXO3A gene polymorphism RS494696 increase the risk of not achieving a molecular response, but not statistically significant through the antecedent variable UTO score. In this pathway analysis, the total effect combining of direct and indirect effect of the FOXO gene polymorphism of the TT genotype increase the risk of not achieving a moderate strength major molecular response. We can conclude that the greatest indirect effect of the FOXO-A gene polymorphism on the failure to achieve major molecular responses is through the high number of blast cells. The results of this study are in accordance with the result reported by Octro et al. 2021, which is stated that genetic factors such as RNX1, ETS, and NBAL2 gene polymorphism were associated with an increase in blast cell and in the blast crisis phase associated with TKI resistance. No major molecular response was achieved. And similar results were reported by August et al. In 2013, which stated that SNP of the BIM C465CT gene was associated with disease progression, blast cell count, and imatinib mesylate resistance. And a report from Willem et al. In 2015 proved that the minor allele A of the BCL gene RS3943258 was associated with increased expression of BCL2 and associated with increased blast cell count and imatinib mesylate resistance. Different results were presented by Ferry et al. In 2019 in contrast to SNP of the P10 gene RS1257387. There was no difference in TKI response in wild and mutant alleles. And Hemot's reports also gave different results. The TET Gene SNP RS344252 homozygous was not associated with difference in blast cell number and response to treatment. And for the conclusion, the study proved that TT genotype or T allele increased the risk of not achieving a major molecular response through leukocytosis, increased blast cell, and advanced clinical phase. The greatest indirect effect of the FOXO3A gene polymorphism on the failure to achieve major molecular responses is through the high number of blast cells. Thank you. Assalamualaikum warahmatullahi wabarakatuh.
Thank you very much for the last speaker today, Ibu Cynthia Oktia Wardani. Okay, for the next one, we will have discussion session for about 10 minutes. Basically have time to this discussion. So if you have any question for our last speaker, three last speakers, please ask the question directly to Bapak Kumboyono, Ibu Ika, or Ibu Sinta Oktia Wardani. Okay, if there are no questions, maybe I will ask some question because I'm interested in the research in Ibu Ika Octaviani. Ibu Ika still here?
Yes, I'm here, moderator.
Okay, thank you, Ibu Ika. I think it's interesting that you have used training, swimming training to the rats.
Yes.
Is it right?
Yes.
For the rats, for swim training.
Yes, swim training.
So swimming training for the rats is only for pregnant rats or for before they become pregnant and then pregnant and you see the behaviors, or how do you explain this training?
Okay, thank you for the questions. Our experimental are 6 group. There are G1, non-pregnant, negative control, no swim. And then G2 is pregnant rats with saline physiologies. And then G3, pregnant rat induced relaxin. And G4, pregnant rats with relaxin, with swimming train. And G5, pregnant rats with saline physiologies and swim train. And G6, non-pregnant rats with swim trains. 3 groups with swimming training.
Okay, so it means that maybe if we compare or we can reflect to our life, so we have to be active or to be exercise.
Okay.
For human, if we are pregnant or something like that?
Yes, the rat. Okay, 1 minute. The rat will surround the swimming medium with a circumference of 74 times 53 centimeters, which is 201 square centimeters. 15 minutes can circle the medium 4 to 5 times. Stop the rat if the rat shows the behavior of sinking its mouth 4 until 5 times. The exercise, core swimming, 4 days, and then rehat 24 hours. And then 4 days swim train again. And the end at 20 pregnancies are stop swim trains.
Okay, thank you so much for your explanation. I hope your project is successful and also your study.
Okay, thank you, moderator. Thank you, Miss Anna.
Thank you so much. So, Bapak and Ibu, I think this is the last of our session in room C. Thank you very much for all your attention and your attendance to this room C. If you still here, maybe let us take our picture. If you still here in this room C, or if you're not you available, you can turn on your video so we can take our picture together. Okay, I will start counting. 1, 2, 1, 2, 3. All right, thank you very much. I can say wassalamualaikum warahmatullahi wabarakatuh. See you tomorrow.
Warahmatullahi wabarakatuh.
Warahmatullahi wabarakatuh.
See you tomorrow and thank you once again.
Thank you.
Thank you.
Thank you.
Thank you.
Thank you.
Recording stopped.